from Pablo Kovalenko that will talk us about Belgica antarctica, this insect as a natural model organism in Antarctica. Now we will try with Pablo's direct
Hello, everybody.
Hi, Pablo. Can you share your screen?
Yes, one moment, please.
Okay, it seems to work. If you can put it as... Okay, you can go. Thank you very much, Pablo.
Okay, just one question. Is my sound okay?
Yes, it's okay. We heard you and we see the presentation. No problem this time.
Hello, dear colleagues. Let me introduce work of our team, Belgica antarctica, as a natural model organism in Antarctica. As Antarctic terrestrial ecosystems host a relatively small number of species. As a result, they are extremely sensitive to climate changes and other influences. Belgica antarctica is a hydromid niche. It's endemic to western shore of Antarctic Peninsula and adjacent islands. Is particularly interesting, especially in the context of this potential use as a model organism for studying the influence of climate changes. This species is very common for bryophyte marsh and carpet formations, for example, from Vanstorfia, Sanionia, and Bryum. Also, you could find this in tall moss formations, nests of some birds like Larus dominicanus and green algae subformation. Also, it's necessary to say that this the area of this species was studied more than 40 years ago. So during these dates, unfortunately, we can't say what's happened with area during this 40 years period. And our team tried to answer this question, and we collected material from more than 300 locations during 14 years period, and known that this species was found. Unfortunately, we not found this species on near the Adelaide Island, and also we have found this species on King George Island. Till our work, there were no information due the appearance of species on this island. So it looks like this species have some changes in their distribution, but we still need more information. Also, for all model organisms, it's very necessary to possibility growth it in lab conditions. And first step for Belgica antarctica was made already. Our colleagues make this for not for all life cycle, but from several stages like larval, and also they get eggs from imagos grown in lab conditions. And also they described life cycle of this species. At least it lasts for 2 years, but they not mention the pupa stage. And for small chironomids and diptera, it's well known that there are also pupa stage, and also this stage were described in very first works on Belgica antarctica in first decades of 20th century. So we collect pupa from several islands, several locations, and make first photo of this stage. Another, since they're not yet discarded, according to this species, is possible sexual dimorphism, because some works belong to this species said that there are sexual dimorphism in, for example, first instar of larvae, and in some cases the works say that there are no. And we try to decide this, try to answer this question, and use 140 individuals from 7 locations, but unfortunately from 1 island. And using principal component analysis, we try to combine all our morphometric analysis. We get momentum length, 5 morphometric analysis parameters, sorry, we analyze, but not found the sexual dimorphism. We suggest that it may be due to different environmental conditions and possibly different genetic features of different populations. And also it's interesting that all studied samples and according to our dates, they belong to 1 population. Also, we described external morphology of Belgica Antarctica and found some malformations on structure of mentum and number of teeth of mandibles. In another hieronymids, the reasons for these malformations could depend from both genetic features in different populations, but most from the different environmental conditions, like different content of some chemicals, for example, heavy metals and oxygen level in substrate. It's very necessary to mention in context of genetic and cytogenetic opinions that this species have diploid set of chromosomes. That's diploid number of chromosomes is 6. It's like in all another Orthocladina subfamily of hieronymids, and this species also has polyten chromosomes, which you could obtain from salivary glands of larvae of this species, and there are some informations that there were some inversions in chromosomes were described in 2 previous works. The late of it was more than 40 years ago, so we decided to know if these inversions are still present in populations of Belgica Antarctica. And we found them. We found inversions in chromosome 2nd and 3rd, and these inversions also were reported in previous work. Also, according to the analysis of cytochrome oxidase 1 subunit gene sequences of Belgica Antarctica mitochondrial DNA, this species is clearly grouped, divided into 4 haplogroups. However, the possible dynamics and changes of them in population of this species is also interesting, because there are only 2 works before us concerning this sequence. And we also analyzed samples. We extract DNA from imago of Belgica Antarctica and get fragments all lengths 700 base pairs. And after analyzing, we see that in studied populations, we studied populations with found that all sequences belong to haplogroup D, and also in one individual were one substitution unique in position 598. Another very necessary thing to model organism is presence of already sequenced genome of this species, and Belgica antarctica has already sequenced genome. And also it's very interesting that the genome of Belgica antarctica is the shortest genome of all studied yet insects. Nevertheless, the size of coding sequences is the same like in another insects. The decreasing of genome was caused due to elimination of length of transposable elements, introns, and another not coding sequences. Also, as this species is very isolated and live in some very unique environment, it was very interesting to analyze the microbiological endosymbiosis which connected and associated with this Belgica antarctica. In pooled seq samples, we detected 44 microbiological sequences of different species, and also we continue work of them, and now is analyzed more samples. So due to these facts, we suggest to use this species as a model organism for studying both the influence of climate changes and the studying of organisms in extreme environments. Thank you for your attention.
Thank you very much, Pavlo. We have some time for question. I don't know if someone want to speak. I can give you, you can open your microphone if you want to ask a question. During that, Pavlo, if you can turn on your camera.
Yes, one moment, please. Yes.
Okay. And I would like to ask you about the, if you or in general, there's some evidence of demographic change with climate change in this species. I mean, the increase in population size, and also some evidence of geographical expansion of the species with climatic change we observe in the Antarctic Peninsula. Pablo, can you hear me?
Yeah, sure. Just I try to turn on my camera back. Yeah.
No, so you can answer without camera, no problem. Okay, here we are.
Sure. Thank you.
So here is my question.
Yeah, could you repeat, please?
Yes, if there are some evidence of demographic change, probably increase in population size in Belgica, and some evidence of geographical expansion with climatic change we observe in the Antarctic Peninsula.
Unfortunately, so grounded works were not found, but at least I know that Poulin and colleagues have described like from several islands near Palmer Station, the destiny of Belgic Antarctica. And as now we are working in also with in this direction to know in which conditions of in which subformations and in which maybe different geographically different places possibly like at least destiny of this species, because very low data, so it's very hard to say about the changes of it, unfortunately, yet.
Thank you, Pablo. Sergey, you had a question for Pablo? You should open your microphone.
Yes, I just a question shortly. Pablo, you discovered some bacteria associated with your stuff. I mean, you just showed us 3 species of 44.
Yeah.
Each are others.
We not included them all, but many of them were like cosmopolitics bacterials, and unfortunately I can't say all you list of these species, but if you're interested of this, you could leave your email. I'll send you on mail.
Okay, and another question, which region of 16S RNA genes did you sequence for bacteria?
Yeah. We on Argentine Islands. It's near Academic Vernadsky Station. It's Fanfare Island. It's like many 3 kilometers north to Galindes Island. It's Wilhelm Archipelago near Kiev Peninsula. And,
okay.
and another population were from Kiev Peninsula. It's called H Hill. It's 5 kilometers through French Peninsula to Galindes Island.
Okay, merci. I will contact you by email. Yeah.
Okay.
Thank you very much, Pablo. And Sergey, you stay here because you are next speaker. And
yeah,
very interesting to see where the bacteria fails to colonize this world.
Yes. Paula, could you please unshare your screen?
Just a moment, please.
Okay, Sergey.
Yes, just a moment. Yeah, just a moment. Yes. Just a moment. Yes.
Okay.
So my okay. Thank you for inviting me to give this talk. It's my talk deals with microbial DNA or microbial communities in the subglacial Antarctic Lake Vostok. Very famous Lake Vostok, and we work only on DNA, not on culturing of bacteria, and use first we used Sanger, but now we switch it to Oxford Convey sequencing technology. I am from Russia. This institution is written above. I am head of laboratory of cryoastrobiology in Russia. Firstly, how many lake and lake-like features under the Antarctic ice sheet? A lot. Last inventory, January 2022, discovered more than nearly 700 such lake and lake-like features. Among them, Lake Vostok is here. West Antarctica is very big one and very famous. I just show you one slide of the Vostok, Russian Vostok station, far away from the coastal area, elevated 3.5 kilometers above sea level, and beneath this station there is a big subglacial Lake Vostok, 200 meters deep sea level. And the temperature above is very low. You see here up to 90 degrees, minus 90 degrees of centigrade. And just 2 slides about Lake Vostok settings. It's a huge lake. In fact, you see here the parameters of this lake, 2070 by 70 kilometers in size. This is a cubic kilometers area. This is not quadrat kilometers. This is cubic kilometers of water volume. It's buried beneath the Russian Antarctic station, beneath up to 2 million years old, very thick ice sheet, up to 4 kilometers, isolated from surface biota. This place in East Antarctica, at least for 14 million years old. So it's ancient in these terms. And until now, no any hydrological links are discovered from the Lake Vostok to other lakes. About the life, possible life in the lake, because our aim to find is there life, which forms of life exist there. Could be very friendly environment. Why? Because the lake, of course, contains free liquid water. Of course, it's deeply buried in dark, 4 kilometers deep, but it's nothing for bacteria. The pressure is up to 400 bar, it's nothing for bacteria. The temperature is perfect for bacteria, so especially psychotrophic bacteria, -2, -3. But the lake contains very low organic carbon content, less than 10 ppb of carbon. It means heterotrophic bacteria alone could not thrive in the lake, only as possible compensals on the autotrophic bacteria. And the main feature which could restrict life there, the lake water is likely very oxygen supersaturated. The values which we have now, calculated values, not measured still, around 800 milligram per liter of dissolved oxygen. I would say in normal condition in cold water in Alps, it's about 14, not 800. So, but bacteria are sensitive, of course, to oxygen because it's a perfect oxidizer. And what we believe what we find out, what we could find out in the Lake Vostok, it should be very special stuff, unique stuff, unusual stuff. In life form, we presented in life forms which we never encountered before. Oxygen life in bacteria is impossible. Extraterrestrial, it's probably possible. Just few slides how we work on this bacteria on this ice, water ice from the Lake Vostok. We work on still ice, frozen water lake. I show you here the cold room in our French laboratory, GCNRS UGA, -15 cold room facilities, and you see here the real photo of the accretion ice of the Lake Vostok, and we use the saw to decontaminate, surface decontaminate the ice, and the polarized light to see the crystals. You see here this polarized light picture, and this is a big monocrystalline ice, and this is a border, and this is another crystalline ice. And after that, we continue to work in clean room facilities of the same laboratory, e.g. CNRS UGA class 10,000, but inside this class, we have a special hoods, this one and this one, class 10,000, class 100. It means we work as cleanly as it's possible. And in addition to this, of course, trying to work on DNA, we run comprehensive, very strict biological controls, which controls, namely sham, mock DNA extraction, negative PCR, ice core wash water, etc. And we established our own contaminant database. At present, this database contains more than 300 sequences. Less phylotypes is less, but this is sequences of 16S RNA genes, bacterial RNA genes. This is very complicated slide. I'll just show you that it was 5G borehole which finally reached the Lake Vostok. It started to drill in 90, and the first unsealing the lake, penetration or enter the lake was happened in February 5, 2012. It was under compensation of mining pressure, and water of the lake rose up and got frozen inside the borehole. And it was redrilled later, in a year later. And the second penetration happened in 3 years later, in January 2015. Namely, from this second unsealing of the lake, I present you data for Vostok borehole frozen water from the depths 3791 meters. This is a picture of this ice, incredible picture, because you see this moon-shaped ice. Why moon-shaped? Because redrilling of the frozen water, the drill biased a bit and entered to the accretion, natural accretion ice. This is frozen water, which rose up in the borehole and got frozen there. This is a picture of this ice, and we of course worked on the lake water, not accretion ice, but this one. Just a moment, I'm sorry. Yeah. Firstly, we run Sanger. Sanger, why? Just to look which diversity are there, microbial diversity are there, because Sanger is very good technique for this. And we sequence it, we of course amplified and sequence it. All DNA extraction and amplification was done in clinical condition, under the clinical condition. We used this region. The region is V3, V4 of 16 RNA genes. We just analyzed 30 clones. Among them, we discovered 16 phylotypes, a huge diversity. Library coverage, Sanger, library coverage by Sanger was just 55%. But of these 16 phylotypes, only one, namely this one, passed all our contamination controls I showed you before. And what interested, this clone, this phylotype, not clone, comprised 40% of all clones by Sanger, and showed very low similarity, very low, less than 90%. It's a level of family, not genus, not species, but family. Very low similarity with Mucilinibacter of bacteroides. We were happy at that time because we considered, we discovered really unknown species in the Lake Vostok, but 15 were contaminated. Then we switched to nanopore, Oxford nanopore. I just show you the data of quality control. This is our sample 3721. We performed base scoring under higher accuracy conditions, streamed barcode, and so on. And finally, we got more than 20,000 reads. Not 30, but 20,000 reads. And you see here, the accuracy of this run was 93%. It's very good for nanopore, but only 1/3 of part of these reads were classified using EPI2Me software by Oxford nanopore. For the first time, we use it for nanopore. As I understand, we are the first. We use it control for nanopore sequencing. What does it mean, control? It means it was done mock DNA extraction, negative PCR, and we tested all reagents in parallel with the real sample for nanopore. Disregarding, despite we did not get any sharp bands by agarose phoresis, we sequenced with nanopore, and we got about 4,000 reads. With the same accuracy level, this is the ME9 we use it, and of them, half were classified, half was unclassified. If we are talking about the species, abundance of species, we use the level, the threshold 0.5 abundance of microbial presentation. Above this level, we discovered only 21 phylotype of bacteria. We were happy because of bacteroides, mostly proteobacteria, actinobacteria, and firmicutes. This is main results, our main results. You see here the names of these species, 21 here. And you see here the reads, the percentage of these reads. And this is a real our sample. This is control sample. Nothing of DNA, real DNA, only reagents, sham DNA extraction, negative PCR, and so on. And we compare this data with this data. If you can see, mostly our findings mostly overlapped with control nanopore sequencing, except these red filled boxes. No, no, no. But when we added here Sanger data, I told you 16 phylotypes were discovered, 15 were contamination. And you see here, no.
Sergey, you should conclude soon, please. We are
yes, thank you very much. I see 3 minutes I have, yes. You see here Sanger, no in nanopore control, but Sanger showed contamination. No nanopore control, but Sanger showed contamination. Only this Psychobacter immobilis showed no here, no Sanger, but it's well-known species of human clinical source, infection, and so on. So our conclusion, all 21 phylotypes above 0.5 abundance level, contamination, despite of they look very, very good. And the last one, you see, this is the name, the species which we discovered by Sanger and considered this is a unique species for the lake. Less than 90% of similarity. But still in the control nanopore, we got 5 reads. Not many, but still 5 reads. So we consider that this is contamination as well. This is data of Epi2Me software, but of course it's possible to use another software. This is another software. You see here the name of this software, and we got a lot of stuff here, but the similarity run was not sufficient for this. Nearly all were classified, but how to decipher contaminants from this, I don't know yet. We are working on this. The conclusion. Any life in the uppermost layer of the Lake Vostok water body is the question. Why uppermost? Because water rose up from uppermost layer. The depth under the borehole is about 80 meters, 8. And we considered that we discovered dominant species, this one. No, it's proven by nanopore sequences to be contaminated. Conclusion, Sanger is rather good, Oxford is better, but more prone to contamination. And key takeaway messages, the subglacial Antarctic Lake Vostok seems to be very likely, highly likely, microbial DNA free. Oxford nanopore outperforms Sanger, of course, but much less accurate, prone to contamination, and not yet suitable for searching for unknown extraterrestrial life, let's say on Europa or in Mars, etc. But on ISS, it works very well. Thank you very much for your attention.
Thank you very much, Sergey, for this talk. We have no time for the question, but we will let your email on the chat. And if people want to contact Sergey and ask question or more, they can do it. We have so to move on the next talk. Will be Volchev. It's quite difficult to pronounce your number. We're looking on Google to do that. We have no idea. Maciejewski, and he will present us the talk "Evolution and Dispersal of Atlantic Foraminifera in the Southern Ocean and Cenozoic Climatic Changes." Over to you.
Yes, thank you. Okay, can you hear me okay?
Yes, it's okay.
Okay, great. Thank you very much. So I'm Wojtek Majewski, and I will tell you about evolution and dispersal of Cassidulinidae. This is group of benthic foraminifera, and these are from Southern Ocean, and try to match it with Cenozoic climatic history in the area. And this work was done in cooperation with colleagues from Geneva, Jan Pawłowski and Maria Holzman, from Łódź, Poland, Aneta Majda and Tomasz Mamos, and from Southampton, this was Andy Gooday. Okay, so benthic foraminifera are a major component of meiofauna. They are highly responsive to environmental change and have good fossil record. Cassidulinidae are rotaliid benthic foraminifera. They have multiple chambers and they are calcareous. They are distributed worldwide, but they are especially abundant in polar regions, and this is where they serve as paleoenvironmental indicators. And that's why they are important for different studies. And so the initial goal actually of this study was to better understand species that are presented in Southern Ocean. And on this picture here, you can see some images from Antarctica. I will focus today on Globocassidulina and Cassidulinoides here at the bottom. Okay, for this study, we used most of all molecular data, and we used a fragment of source unit ribosomal DNA. It actually included a barcoding fragment that was used for taxonomy and phylogenetic studies for a few decades now for this foraminifera. We had altogether more than 100 isolates or individuals, and they provided us almost 200 sequences. Half of this material came from Antarctica. These were most of all the Ross Sea and Admiralty Bay in South Shetlands, as well as Rotterdam. Then the second half was from outside Antarctica. I will call it sub-Antarctic. This includes South Georgia, the Falkland Islands, and southern Patagonia. This was mainly in the area of the Beagle Channel. These are results of species delimitation. So with different color, you can see different taxa. We used 6 different delimitation records, methods. I don't know if you see the top of this picture. Oh, sorry. Oh. But there are these methods indicated there. Maybe you don't see it because I have a line here. Okay, so the most complete data we had for Cassidulinoides marked in yellow, and for Globocassidulina marked in blue, and in both cases we had 3 species. These are maps showing distribution of these species, and these distributions or ranges are based on our molecular data. So on the left you have Globocassidulina, and here the pattern was pretty straightforward. So we had 2 Antarctic species. One was slightly deeper water, and the third species was sub-Antarctic, and there was no signs of gene flow across the trade passage. And we could actually use morphology to discriminate between these 3 species. In Cassidulinoides, the pattern was much more complicated. So we actually had little match between molecular species. These are marked by colors here, and morphotypes, which are marked by hatching. And this was probably due to interspecies hybridization that took place sometime in the past. So with having this result, we could actually say, okay, we know now the species, so the job is done. But we actually wanted to use this data set, molecular data set, for getting as much information about evolutionary history of those things and maybe dating some events as possible. So for this, we used Bayesian time-calibrated reconstruction. And for calibration, we used, of the molecular clock, we used 2 well-dated events from closely related rotaliid foraminifera. These are marked by letters A and B here at the bottom of this graph. And in 2 cases, which are marked by these red cycles, we actually verified these results with fossil record. So this fossil estimate of these events is marked here by green labels, and this gave us actually pretty good match for Globocasidulina, but not so good for Casidulinoides. This is the bottom example here, and this also indicates that the dates that I will be discussing later on, which are based on molecular data, they should be somehow younger, in fact. Okay, so these are the same data that I showed before, but now we are focusing just on these two species, I mean genera that are of interest. So the two conclusions from that is that diversification of both Casidulina and Globocasidulina and Casidulinoides from the Southern Ocean began sometime in the Miocene, and it was long after the Eocene Oligocene cooling and initial opening of the Drake Passage. And the second conclusion from here is that this diversification continued through warm and cold phases of climate during the Neogene. And these phases are marked here in yellow and blue. If you are now talking only about Globocasidulina, there were several interesting events, and the first one took place exactly 15 million years ago. It was a split between Antarctic and sub-Antarctic lineages that took place during the mid-Miocene warming. And since then, there was no gene flow across the Drake Passage. The second event took place about 10 million years ago, and it was diversion in Antarctic species of Globocasidulina. And it seems to be actually associated with bathymetric structuring of water column that was driven by intensified production of cold Antarctic bottom water after the mid-Miocene cooling. This species, Globocasidulina subglobosa, is actually now deeper water than the other one, and actually based on fossil record, it looks like the ancestral one was rather shallow water than deep water. Then we have this event here about 5,000 years ago. This was probably an episode of gene flow between South Georgia and Patagonia, and finally, slightly after 2 million years ago, we have final diversification of Antarctic species, and this was probably in conjunction with a glacial intensification that took place at that time. In Casidulinoides, we had 3 episodes of active gene flow across the Drake Passage, and they all seem to correlate with warm climate phases. The older one was here, and probably it was also related to mid-Miocene warmth or warming. And based on fossil record, it looks like there was a transfer of Antarctic species into sub-Antarctica. The younger ones taking place probably during the late Miocene to early Pliocene warmth. These were the hybridization events that I mentioned before, and actually they indicate that there was probably a transfer of species from sub-Antarctica to Antarctica. So after this finding, we could conclude that climatic warm phases, in this case the mid-Miocene warming and the late Miocene to early Pliocene warmth, they are likely to be the key periods for biologic dispersal across the Drake Passage. And actually these were important events that shaped the present day biodiversity in Antarctica. And this observation stays in line with recent dispersal across the Drake Passage, diminishing of environmental gradient between sub-Antarctica and Antarctica, and reconstructions of warm early Pliocene. This is actually not all. We also have this characteristic star-like pattern in many of those foraminifera. This is shown by these haplotype networks here at the bottom. And they are interpreted as echoing bottlenecks followed by episode of demographic expansion. And they were actually dated, and these demographic events were dated at about 400,000 years ago. And it looks like this date was not accidental. At this time, there was a especially long interglacial that you can see here in the record of atmospheric CO2. And also there are some indications that actually the grounding line in East Antarctica in Wilkes Land, this publication, was several hundred kilometers at the time more to the south than it is today. So just simply the living space for those things about 400,000 years ago was much larger than it is even today. And conclusions. There is more species of Southern Ocean Cassidylidae that we previously anticipated. Their molecular species do not always match with distinct morphotypes. And more general ones are that the diversification of Southern Ocean Cassidylidae began in mid-Miocene. Climate warm phases seems as the key period for biologic dispersal across the Drake Passage, and this is, I think, the most interesting outcome of this study. And the final one, demographic expansion in Southern Ocean Cassidylidae predates the last glacial maximum and seems to date back to the long interglacial about 400,000 years ago. This is the founding, and these data are already published in this publication. Thank you very much for your attention.
Thank you very much, Wojciech, for this very interesting talk. We have some time for a question. If someone want to ask one, I can open your microphone to speak directly, or you can write it on the chat, or in there is question answer section also. So we have plenty of possibility to ask some question. Waiting, I have some. Wojciech,
yes.
When you present this lot of different networks at the end, we see that generally Patagonian and South Georgia species share some genetic haplotypes. And it's quite surprising that South Georgia is more similar to Patagonia than to Antarctic locations. Because for a lot of marine species, we see more affinity of Antarctic and South Georgia than South Georgia to Patagonia. Probably, and the interpretation is the presence of polar front between South Georgia and Patagonia. So I would not expect to see this kind of pattern of connection between South Georgia and Patagonia in these pelagic groups.
I mean, these are benthic foramifera. I maybe forgot to answer. These are shallow water benthic foramifera. They have a propagules that can be in water column, but basically they are pelagic. But it's actually not in different groups. You could see that actually in these two closely related genera, actually the distribution pattern and the history was quite different. So probably it's also important. The important factor is also ecology of those things. Yeah, in Globocasidulina, for example, there was no gene transfer between Antarctica and Patagonia for the last 15 million years. Yeah, so it looks like there was actually a barrier, and South Georgia at the time was not an island yet. Yeah, so that could be a factor. In Cassidyllinoides, it's much more complicated. Yeah, but okay. there was a transfer, yes, between Antarctica and subantarctica. So actually, the species, one species of South Georgian Cassidyllinoides looks like it is endemic, but it's actually just one morphotype. But it seems to be endemic for South Georgia.
Okay, thank you very much. I don't know if there is more questions. No.
We are almost on time.
So we are on time. Thank you very much, Wojciech.
Thank you.
For the next speaker, it will be Daniela Monsanto that will present us landscape genetics of the springtail endemic to Marion Island. Is Daniela here?
If it's sharing shows. I'm not sure what you can see right now.
We don't see in presentation mode.
Okay, let me just try again. See that screen. Still nothing?
No, still not. Oh, okay. Okay, Daniela, you can go.
Okay, perfect. Good afternoon, everyone. I'm Daniela, and I'll be presenting a component of my PhD, looking at landscape genetics of a springtail endemic to Marion Island. So, understanding the interplay between genetics, species biology, functional ecology, and landscape ecology in driving organismal evolution has become central to answering many fundamental questions in evolutionary biology. And this can provide insight into historical as well as contemporary processes. Now, specific for this talk, we'll focus a bit on landscape ecology, which has been defined as the study of the pattern and interaction between ecosystems, the manner at which these interactions affect ecological processes, and specifically by incorporating the effects of spatial heterogeneity. So just to give an idea of, just for illustrative purposes, landscape ecology is essentially the interplay between landscape heterogeneity as well as intrinsic characteristics. So for instance, in this fragmented habitat, we have perhaps a natural barrier as well as a man-made barrier, and organisms that have high dispersal capabilities would, or perhaps could not be directly influenced by landscape barriers. So for instance, a white-plumed honeyeater could potentially overcome patchy habitats or fragmented habitats, and potentially even a landscape barrier such as a road. Yet an organism with poor dispersal capabilities such as the eastern fox snake would likely be constrained within their habitat matrix. And the manner at which these organisms interact with their landscape as well as neighboring populations influences genetic structure. So in terms of population dynamics, just for instance, just as an example here, if we have 4 populations which would interact with one another, and this interaction would give us a structural pattern. Within each of these populations, we have smaller intra-population interactions, and furthermore, we have additional interactions with neighboring populations. And what's interesting is that depending on the scale at which you focus on would provide a different structural pattern. And this would be the case if we were to look at the sub-Antarctic region as a whole, or focusing mainly on an island or even a fine scale 1 kilometer length transect. So structural patterns would obviously be affected by the scale at which you focus on. So specifically for landscape genetics was first described in 2003, and it's essentially when you overlay genetic structure onto a landscape. Since then, several studies have been published talking about the topic, and a multitude of studies have been conducted looking at various organisms. So landscape genetics pulls a broad range of disciplines or fields. And in order to provide robust landscape genetic analyses, we would require input from a few of these fields. So specifically to Marion Island has been characterized by its previous historical events such as volcanic and glacial maxima or processes. We know in the sub-Antarctic region, these islands are exposed to quite harsh climatic conditions, and both of these factors drive the heterogeneous landscape that's present on the island. And this heterogeneous landscape would affect or shape biodiversity patterns on the island. In addition, and as to what I mentioned previously, the organism itself, the dispersal capabilities of the organism, as well as habitat preferences of the organism would influence biodiversity patterns. In this case, we're focusing on a springtail, which is Cryptopygus antarcticus trevei, which is a springtail endemic to Marion Island. There are about 1 to 3 millimeters in length and are found in large abundances on the island. So I mentioned a little bit earlier about scale, and scale needs to be understood from the perspective of the organism itself. So assuming a large organism traveling a distance of about 5 meters, it's not such a big deal. But if we scan a landscape and focus on something a lot more microscopic, 5 kilometer distance is quite a daunting task to overcome. So previous research on Marin Island has been conducted looking at large scale, or looking at the scale of the island for the flightless moth, Pringliophage marioni. And they found that there were 3 genetic clusters located on either side of ecological barriers. So this barrier being a climatic barrier, and this barrier being a geological lineament across the island. But on a fine spatial scale, only one study has been conducted looking at about 1 kilometer length transect, and they found that on an Azorellus alogocushion plant, and they found that within just 10 meters, species, or organisms or individuals were genetically different. And this just shows that looking at a really fine spatial scale, we see a completely different picture to large spatial scales. This then brings us to our research aims, where we aim to explore the genetic structure of a springtail endemic to Marin Island. We identify the landscape features that can facilitate or impede gene flow, and we also identify the role that environment plays on shaping genetic structure. The fourth aim here is not included, but is coming soon. So that's just looking at the direction and drivers of gene flow. So this is just an idea of the workflow. So it's comprised of field work, looking at 2 study sites, which I'll show in the next slide. It then had followed by lab work, which involved DNA extractions as well as microsatellite genotyping. The necessary software was used to look at population structure, and finally the landscape genetic pipeline was followed. So these are the study sites on Marin Island. I have 2 transects, Nelly Humps, as well as Skua Ridge, and you can see that the scale at which we're working with, we're looking at a 1 kilometer length transect for both of these regions. So I'll start off by speaking about Nelly Humps, the 1 transect, and then present the results and then move on to the 2nd transect. I do, however, focus a lot more on Nelly Humps due to time constraints. So this is a sampling site with all sampling points, 2 sampling transects that are perpendicular to one another and are presented here on a digital surface model, just to show elevational changes. And to incorporate the landscape genetic component into the study, our collaborators drew up polylines between each sampling point pair to then calculate the Euclidean distance as well as the surface distance, and then in addition to that, terrain ruggedness and surface complexity. So to speak a little bit about the results and looking at structure plot or the population structure and overlaying that onto the landscape, where each segment of these pie charts corresponds to the population membership to a specific cluster. And what's interesting is that, and I'll be focusing on this area here, we see that there's a distinct genetic break that's occurring at this position. We have this light blue cluster in contrast to the gray and the blue clusters on the bottom half of the transect. And located at this point, which was photographed on the island, we have a 3 meter high ridge. And remember, I spoke a little bit about scale, and for a microscopic organism, a 3 meter high ridge is quite a big obstacle to get through. But if we're looking at the panchromatic imagery of the area, which highlights the lava flows of the area, I'm now encircling a black lava flow, which is of different geological age to the neighboring area. And what happened here was there was some form of volcanic eruption, and the lava flowed around this highly elevated area, obviously resulting in the extinction of species within or individuals within the direct line of lava flow. And this is the driving force behind this genetic break that we're seeing. In addition to this, the lava flows alter the geophysical or the physicochemical properties of the soil, which would then also influence the vegetation type. So in the demarcated area, we have more drainage line vegetation such as bryophytes and grasses. Yet in the surrounding areas, we have your fern breaks, your vascular plants, and a very rocky landscapes. So the next thing was now to incorporate resistance and terrain roughness. So we used the terrain roughness as a direct input for resistance, and a high resistance value here would mean minimal movement or resistance to movement, whereas a low resistance would be ease of movement amongst sites. So the software that we used here was CircuitScape, and it essentially looks at conductance and voltage and assigns a value to each grid, to each cell on this grid. These values were the terrain roughness that I've just mentioned, and we then simulate roughness, or I mean resistance, across the landscape to see the features that would influence movement. So when overlaying this resistance map onto the landscape or the digital surface model, we can see that high resistance, being the really bright colors, corresponds to these highly elevated areas, as seen here. And this just shows that the changes in elevation across the landscape do indeed provide resistance to movement, which is what's expected. Additionally, we looked at that barrier that I mentioned earlier, where we had a genetic break happening here, and I associated a binary variable to individual interactions across these this barrier. So individuals from site 9 and 93 were assigned a binary variable of 1 to say that there is a barrier between these points, yet site 1 and 31, I assigned those interactions as 0, which means no barrier. And using generalized linear models and comparing kinship coefficients, which is the genetic metric, to the landscape metrics that we've got, we then compare kinship coefficient to the Euclidean distance between each sampling point and each individual. And the same goes for surface distance, landscape resistance, and so on. And the most likely model based on the Akaike information criterion is resistance. So this just shows that resistance, landscape resistance, is indeed the driving force for the genetic patterns across the island. And second to that would be the barriers. So the landscape barriers that we see on this landscape would be the second most driving force for the genetic structure or genetic patterns on the island. In terms of the second transect that we're focusing on, we're looking at Skua Ridge, which is a little bit more heterogeneous in appearance. We have a grey lava land flow here, as seen by the Google imagery there. A grey lava land flow, we have black lava on either side of it, and then also a major stream or river that intersects the transect. And this was so that we could then test different landscape features, you know, and to identify their effect on the genetic structure. And just briefly, I don't go into too much detail on this transect, but just briefly, we can see that genetic structure is potentially not as clear-cut as initially thought, but we do see some genetic structure that broadly coincides with the landscape. So we have here, like the individuals here are predominantly from the dark blue cluster or cluster 1, yet individuals on the grey lava flow are likely members of the grey cluster. So again, not such a clear-cut result, but we do see that there is genetic structure that coincides broadly with the landscape features on the island. I just want to draw your attention to this area here, as well as this area here. We can see that there's a mixture of all groups, but when looking at the resistance across this landscape, we see that the resistance coincides with these major landscape features. We have a lot of resistance in this area, which is likely due to the just landscape features or the terrain roughness in that area, which might be a little bit more complex than looking at it at first sight. And then we also have here this the river here, which we see is also quite a dominant driving force for landscape resistance or for genetic structure. And I don't go into detail with the linear models for this due to time constraints, but I just wanted to mention that coming up next, we plan to look at gene flow and the direction of gene flow. And this will be done using the this paper here, which is a group from the University of Pretoria. And we'll use their wind simulation models where they use computational fluid dynamics to simulate the wind movement across our sampling transects. And we can then test for the direction of gene flow and, you know, if the prevailing wind direction is the main driver of gene flow. So what does this mean? It essentially just means that complex landscape does result in complex genetic patterns, and that high resolution GIS-based landscapes are extremely important in identifying genetic structure across landscapes. We know that landscape resistance and barriers shape structure on the island, specifically for the springtail. And springtails can be used as proxies to understand or study landscape genetics. The last point here is that fine scale work is extremely important, as we see that fine scale work shows a completely different picture to what would be seen at the scale of an island. But this can inform us about migratory patterns such as gene flow, the genetic neighborhood, the biology of the species, as well as for conservation plans, where focus should be drawn to fine scale work rather than just broad scale, large scale landscapes. And with that, I would like to thank the following people and organizations, and you're welcome to contact me if you have any questions or suggestions. Thank you.
Thank you very much, Daniela, for this amazing fine scale genetic structure. That's really very interesting. We have no time for question. You use your 15 minutes talk, but we will let your email on the chat. If there are questions, you can also, after the talk, let some question in the question answer section, and Daniela can answer in the same time. So as you have seen, we are going from east to west. We began in Australia, passed through Russia, Ukraine, Poland, now South Africa, and we arrived to Europe with Gilda Cacavo from Italia, I think, that will present us a genomic perspective on Antarctic toothfish circumpolar population structure. So, Gilda, up to you.
Thank you so much. I'll start my screen sharing. Okay, and now, can you see my proper presenter view? The presentation.
Okay, Gilda, you can begin.
Thank you so much, everyone. I'm very excited to speak to you today about Antarctic toothfish population dynamics in the Southern Ocean. As Elie said, my name is Gilda Cacavo. I did my PhD in Italy, but then I did a postdoc in Germany, which is what I'm going to be talking to you about the work for, today. And now I'm actually in Paris, where it is currently 36 degrees Celsius. All of these affiliations down here, the work that I'm talking to you about today was done at the Alfred Wegener Institute in Bremerhaven, the Berlin Center for Genomics and Biodiversity Research, and the Leibniz Institute for Zoological Research, also in Berlin. But I am currently based in Paris, where I'm associated with two climate laboratories under the umbrella of the Institute Pierre-Simon Laplace, as well as Genoscope, a genomics institute here in the Paris area. But talking about the German work today. So, changing gears a bit from the terrestrial to the marine, I'm going to be talking to you today about the Antarctic toothfish, or Disostachus mawsoni. And so for those of you who don't know, Disostachus mawsoni belongs to the Antarctic fish clade of Cryonototheniidae, which we can see on the left. Huge number of fish, one of the few examples of marine teleos adaptive radiation. Very exciting stuff on this group of fish, but I will just be focusing on one belonging to the suborder Nototheniidae, Antarctic toothfish today, as you can see up in the top left corner here. And so Antarctic toothfish are some of the largest fish in the Southern Ocean. They can get up to 1 to 2 meters in length, and they have a circumpolar distribution. We can find them along the shelf, slope areas, as well as in offshore seamounts. And in addition to being of interest to humans, as I'll describe shortly, they're also an important prey species for whales and seals in the Southern Ocean. And so the way that they're important for humans is that they support a $500 million US fishery annually. And what this graph here shows, starting from the 1980s when the fishery began, we see this marked increase in the year 2000 in landings of toothfish generally that leveled out more or less in the past 20 years. And so this brings into the question of why there is particular interest in understanding the dynamics of this fish, because indeed the management, the sustainable management of the fishery requires it. And so, as many of you may already be familiar with, Southern Ocean fisheries are managed by the organization CAMLAR, which stands for the Commission for the Conservation of Antarctic Marine Living Resources. And so in order for sustainable, or as they often say, precautionary management to be successful in the Southern Ocean, we require information about population dynamics, and part of that can be informed by genetic population structure. And so what my project was aiming to look at was how we can inform sustainable fisheries management of Antarctic toothfish using genomics as well as autolift chemistry to test population structure hypotheses. So unfortunately, I won't have time to talk to you about the autolift chemistry part of the work today, but definitely follow up with me about that if you're more interested. And so Daniela gave a great introduction about population genetics and landscape genetics and why this is valuable. My explanation is going to be a lot simpler just to get everybody on the same page. So we can think of the null hypothesis for population genetics being one of panmixia. Different groups of populations are exchanging enough individuals that we can't define differences between them genetically. But as barriers are introduced between groups of populations, be these geographic or related to human intervention, we start to experience population isolation. And so then these panmitic populations become more and more isolated to the point of potential full isolation, in which we can start to distinguish between them genetically, in which we're starting to see fewer exchange between the groups. And the way in which this would be useful information for managers to know is that if we have a situation of isolation, we're losing genetic diversity within these isolated groups because there's less exchange from outside groups, and a less diverse population is a less resilient population to climate change effects, fishing, and so on. And so what my project aimed to do is to test between different population structure hypotheses of Antarctic toothfish. And so the boxes and the green circles represent the areas from which I had samples. And the idea was I wanted to see the extent to which different groups that we could define of toothfish around the Antarctic are more or less connected. And one of the main drivers of this connection I was hypothesizing were two current systems. One is the Antarctic circumpolar current, shown here in yellow, going from a west to east direction, and the Antarctic slope current going in the opposite direction, but hugging the slope of the Antarctic shelf area. And so prior work had basically been trying to test between two extremes of the way population structure could be of Antarctic toothfish: a total heterogeneity of populations around the Antarctic and one of total homogeneity, or this panmitic or panmixia situation that I described before. So work from the 2000s was in support of this heterogeneity of toothfish populations around the Antarctic, while later work in the 2010s was actually more in support of a homogenetic organization. And now recent work that has come out in the past couple of years has shown further support for that homogeneity. However, there are some interesting exceptions to that. For example, in Choi et al. Looking at differences found based on mitochondrial DNA that were not found based on nuclear markers. And so my work here was hoping to contribute to this story and try to understand better what the dynamics are, what is the structure hypothesis that we can more or less support, and how can we use that to make management decisions. And this was on the circumpolar scale. On the regional scale, my focus was in the Weddell Sea for multiple reasons. So the Weddell Sea is an area where we know very little about Antarctic toothfish structure. A lot of what we know derives from the Ross Sea area, where the fishery has existed for the longest time. And I was basically testing between three main hypotheses: one of locally distinct populations, so that's one extreme, one of one large panmitic population over the Weddell Sea, and one sort of Goldilocks middle ground, which represents two populations that are structured around the oceanographic structure of the Weddell Gyre, which has a branch that moves towards the shore in this area here. So in order to do this, my project followed the following course of action. So collecting samples of tissue and autoliths from a group of international collaborators to whom I'm gratefully indebted. DNA extraction and quantification, of course, and then preparation of 2 types of libraries. So 3RAD was the reduced representation sequencing method that I employed amongst different RAD sequencing methods to choose from, as well as whole genome sequencing, which was a sort of exciting new part of the project that was added in during its development. And finally, these libraries were then sequenced on a Novusec S4 sequencer. Oh, and of course, I shouldn't forget the trace element analysis, which unfortunately I can't talk to you about today. And this was performed at the Alfred Wegener Institute. And so something else new about this project that wasn't necessarily planned in the beginning was using otoliths to extract DNA. And I don't necessarily mean extracting from the bone itself. So, right, otoliths are fish ear bones. Bones don't have necessarily DNA in them. But what we're actually extracting is DNA from trace amounts of tissue that are adhered to the outside of the otolith. And we do this using DNA extraction kits that are optimized for low volume elutions, because we're really dealing with amounts of DNA between 0.1 and 1 nanogram per microliter. And I'm happy to provide more information either after the talk or later about the method that we developed here and its success and so on. So in terms of the sequencing methods, we chose among the RADSec methods 3RAD. And this was a method that's been developed at the Berlin Center for Genomics and Biodiversity Research, and it's optimized for relatively low quality quantity amounts of DNA. It generally allows us to minimize processing steps, reduce chimeras, eliminate adapter dimers, and to identify PCR duplicates in the process. And so we did an in silico digestion in order to determine what restriction enzymes we would use to chop up the genome in our reduced representation library. And we ultimately decided that for Antarctic toothfish and for the method that we were using, these 3 enzymes were the ones we had the best success with. So for the whole genome sequencing method, I used a kit from New England Biolabs that enzymatically fragments the genome into lengths of around 500 base pairs. This was a 24 sample kit, but what I managed to do was actually dilute the reagents in it 3 to 5 times to stretch that kit to process 3 to 5 times more samples, which was a nice money saver there. Now, bioinformatically, what we did with the whole genome libraries, which was important because these are low amounts of DNA and not necessarily great quality and subject to contamination, is we made sure to check that we were only using reads that belong to the Antarctic toothfish genome, and we also removed duplicates that were detected in the sequencing and PCR process. So in terms of, and I'm finishing up soon, sorry about the timing, the 3RAD population pools we had derived from these 4 areas circled here in the different colors. So the Weddell Sea, 1 from the Ross Sea as an outgroup. Unfortunately, the Ross Sea samples didn't work. Don't worry, we included them in the whole genome study, and we ended up with 109 samples total, just focusing on the Weddell Sea population structure question. For the whole genome resequencing work, we had 5 circumpolar areas represented, 4 in the Weddell Sea, 1 in the Ross Sea, 1 in the Amundsen Sea, East Antarctica. Unfortunately, in the Bellingshausen Sea, our samples did not work. No endogenous toothfish DNA in those samples, unfortunately. And we were unfortunately unable to get further offshore samples. So we ended up with 48 total samples. So as I mentioned, these were sequenced on an S4 lane altogether. And in addition, the trace element analysis work we conducted on the same 48 otoliths that we did the whole genome work on, getting trace element work from the edge, nucleus, and transects of otoliths. This is a scanning image of a otolith and the laser ablations that we did there. Again, sorry, I can't talk to you more about that work. And unfortunately, the analysis is still in progress. I was really hoping I'd be able to share some preliminary results with you today, but c'est la vie, as we say here in Paris. So hopefully in a coming Congress, I'll be able to share some more exciting results, and hopefully you can keep an eye out for the paper coming out hopefully in the next year. Preliminarily, the coverage looked good that we had both for the 3RAD and the whole genome work. We had most of the 3RAD samples were at a coverage level that was at our goal. Whole genome was a little bit worse, but we're seeing what we can do with the samples that were under 5x coverage. So in summary, just to remind everybody, toothfish, important top predators, managed by CAMLR, commercially exploited. Population structure, there's still some open questions. There's a large body of work that's been contributing to this, and I'm hoping to add a drop to that pool. Current study uses genomics and autolyth chemistry, circumpolar focus, regional Weddell Sea. And finally, employing genomics methods, some new to toothfish, such as autolyth DNA extraction and whole genome resequencing. And I just want to thank my collaborators on this. So at the Alfred Wegener Institute, Thomas Bray and also Kirsten Beyer, as well as members of the AVI Proxy Lab, at the Berlin Center for Genomics and Biodiversity Research, Camilla Mazzoni, as well as Larissa Arantes, who is actually getting married this weekend. So unfortunately, I'm here and not there, but just putting a shout out to her. My email was on all the my website was on all the slides if you want to learn more, and my email is here if you want to reach out to me after this talk. So thank you so much for your time, and I look forward to seeing the rest of the talks in the session.
Thank you very much, Gilda. We have time for perhaps one question. Someone? Nobody has a question today.
Question and answer.
Oh, someone. Why extract DNA of otolith and not tissue, Gilda?
Great question. So obviously tissue can get us better tissue quality and quantity, better DNA quality and quantity. But the thing for many fisheries, Antarctic toothfish included, is that otolith collection is a standard part of scientific observer sampling for stock assessments. This is because otoliths are typically used for aging, and aging information is used in the stock assessment. So the idea is that we have otolith samples going back 20 years for Antarctic toothfish and that are continuing to be collected. So this is a rich sample base, and it is also a lot easier to collect otoliths. You don't need alcohol. They're already being collected. This is much easier to implement in terms of a fisheries management perspective. And with the new things that we can do with whole genome resequencing, we have the capacity to use the sort of poor DNA quantity and quality that we get from otolith DNA extractions. So I really see it in particular in Southern Ocean fisheries as kind of the future of the way in which we can continue management monitoring of genetic structure. And so obviously it's a bit more difficult in terms of the DNA extraction and the subsequent library prep and bioinformatics, but that's just going to get easier over time as sequencing costs go down and as methods are standardized. And if you want to learn more about the protocols and methods that we developed in this project, feel free to reach out. I've got all the protocols.
Okay, thank you, Gilda. A very short answer to the next question, please. Gemma Collins asked about the estimated genome size for this species.
So 9 billion bases.
Okay, thank you very much. It was a very short answer. And so we move to Gemma Collins that will talk about DNA barcoding of Antarctic mites, including spatial separation of free clades of Nanochestus antarcticus. Gemma, it all seems okay to start.
Okay. You should see my screen, hopefully.
Yes, all is okay.
Yeah, it's okay. Okay, yeah, so hi everyone, my name is Gemma Collins and I'm a postdoc at the Leuven Center for Translational Biodiversity Genomics. And TBG, this is like an umbrella organization, including six different institutions that you can see the logos for, and myself, I'm based at Senckenberg in Frankfurt. So usually I'm working on whole genome sequencing for many different types of soil invertebrates, but today I would like to share with you a nice little story about the Nanochestus antarcticus. So this is a really tiny Antarctic mite, and we have some existing DNA barcoding data, and I will show you how the divergent clades are spatially separated. So at the moment, I'm putting together this small manuscript together with Monica Young, based in Canada. So she's really into the taxonomy of mites, also along with Pete Convey and Ian Hogg. So this is the slide that I usually start with because during my master's and PhD research, I was looking at the spatial distribution of CO1 barcodes for Antarctic collembola, also known as the springtails. And these tiny invertebrates have survived in situ on the continent since before Antarctica separated from the rest of Gondwana. And yeah, so they're truly ancient relics and they have several behavioral and physiological mechanisms to avoid freezing. And they are, in fact, the largest terrestrial animal on the continent. And there are 10 species of springtail in the Ross Sea sector. So this includes Victoria Land and the Transantarctic Mountains. But now all of these species have been sequenced. Whereas on the other hand, mites are also ancient relics. They have endured a similar history. They're even smaller. So they do occur in slightly larger population sizes, and there are also more species of mites. But it has actually taken us quite a long time to get to this point where we have enough barcode sequences to form a story about the mites. And of course, the story starts in a similar way to some of the previous talks we've just heard about, picturing this fragmentation across the landscape. So here you can see really obviously this kind of island habitat that's available to these animals. And you can see like some exposed soil, but they simply find it really difficult to make their way from one place to another. And so, yeah, you end up with these like isolated populations, even though they may not be necessarily located that far apart geographically. But of course, when populations are isolated like this, they gradually become more and more different genetically. And so with CO1 barcodes, we have discovered this kind of spatial diversity many times in the springtails. So here are a couple of small examples from 2 species found in the southern Victoria Land region. And at each of these sites, we sampled really extensively. So we aspirated what we could see by eye. We took soil samples, vectorscope base for flotation to extract the animals from the soil. And when we camped for several days in some of these locations, then we also used pitfall traps. And so here you can really see that for the species on the left, Antarctocinella monoculata, there were these 2 main genetic groups that were found in slightly different areas, all in the southern Victoria Land. And for the other species here, Cryptopicus nivicolus, there were these 3 genetic groups. And so we already know quite a lot about the springtails and how we find again and again, basically in every species that we look at, all of the 10 species, that there really is this disconnect between locations, and that's what has resulted in all of this hidden diversity. And so this is what we were kind of expecting to find, or what we were trying to explore for the mites, that they differentiate according to the landscape areas. And there are 15 species of mite, but probably more if we consider this genetic diversity, right? But also, in addition to like this idea of cryptic species, there are already morphological differences that are starting to be found. So for example, in one genus, Stereotydeus, there are 15 species if you include like the whole peninsula sub-Antarctic island area. And in the Ross Sea sector, which is where we were focused, there are even 7 species of this genus. And there's been some recent attention given here where this paper, they basically went through the whole taxonomy for this genus, and they described 2 new species. And this gives kind of an example of the more you look, the more you find, and also that there's a lot of work still to be done in the mites. These authors also sequenced some Stereotydeus and including some of the same locations as us, but unfortunately they were looking at the 3' end of the CO1 gene, not the typical 5' end, so we actually couldn't include the sequences in our alignment. But what we did do was basically to sequence as many mites as we could. And so we had access to samples from many different locations, and over the years we had a fair bit of trouble sequencing them, but gradually, like from selecting from the samples that were taken over 10 years or so, we eventually have now 131 sequences for this paper, and these are coming from 18 different locations. So 2 of the locations are actually from outside of the Ross Sea sector. So this gives a really nice context to the study, and this is thanks to the Antarctic circumnavigation expedition, which was in 2017. So here you can see like this little red dot where we have another location, the Saipol coast. And then we also have some samples from the Macquarie Island. So now just zooming into like the three main areas that we were interested in. So if you go to a particular site in the Transantarctic Mountains, how many species would you possibly find? And the answer is 4 species in this region. For southern Victoria land, there could be up to 6 species of mite from all genera. And in northern Victoria land, up to 10 species. Yeah, so one of these species of Stereotydeus, it was actually one of the new ones that was described in this new paper that I just showed you, is found from both the southern Victoria land and northern Victoria land region, so a little bit of crossover. And there's also 2 other species that cross these boundaries, which are typically barriers to most springtail and mite species, but we suspect that they're probably different species that all currently have the same name, right? That's why we're particularly interested in the Nanocestes antarcticus. And yeah, while we tried to sequence as many different species as we could, we had in the back of our minds that Nanocestes antarcticus is a particularly likely candidate for this kind of hidden diversity. And the other reason why the species is particularly interesting is because it is the smallest species of mite. It's also the most widespread, apparently, and the most abundant species. I mean, maybe this is not as widespread as we think. That's why I said apparently. There could be cryptic species, but there also could be morphological differences that we just haven't looked for yet. So, for example, the Nanocestes antarcticus from the Peninsula region in the 1980s or so, it was renamed and replaced with 2 other species names. So again, like the more you look, the more you find. And it's a shame so far there haven't been any DNA sequences, and so that's why we are focusing on this group. So now I can show you a little bit of the tree. I have this idea now where I'm going to like step you through the different taxonomic groups of the tree, going from the bottom to the top, and then I finally finished with the Nanocestes, which is this separate clade you see at the top. So first of all, a little diversion. So there are 2 Cockerigidia species known from this region, and we managed to sequence both of them. So here you can see that, yeah, there are the two species here. So the northern species, which has maybe 3 groups or 3 potential cryptic species, and the southern species that we have sequenced as well. But I could only find 5 individual sequences on GenBank for this whole family. So even though, yeah, it's quite a simple kind of story, it's still maybe a meaningful contribution to provide these sequences. And then going up the tree, so we have a whole bunch of Stereotydeus, or actually some of them could be Tydeus. There might be up to 9 described species here, and it's a little bit difficult because their distributions are all overlapping. Like I can show you this distribution map from the authors that I was mentioning before, who redescribed the taxonomy for the Stereotydeus genus, and here you can see, like at some of the locations, you have multiple different species that could be found. So yeah, we're going back to the vouchers and taking a closer look at the taxonomy now. So yeah, coming back to the tree, we were up to this Tydeidae family, and next we also have some Eupodes wisei. There's one group on the tree, and they're all basically the same from the Cape Hallett area in northern Victoria land, and we have several other species from this genus on GenBank, so that's kind of nice. And now finally at the top we arrive at the Nanocestes clade. So let's zoom in here, where you can see that we found 6 really different genetic groups. So 6 clades represented in the boxes here. And these cover all of the major terrestrial areas in the Ross Sea region, as well as those 2 additional locations, the Saipol Coast and Macquarie Island. So 2 things stand out to me here. One was that in the Transantarctic Mountains, so there's like darker blue boxes, we had 2 potential species occurring in the same locations. And the second thing that was interesting to me here is that one of those groups is also the one most closely related to the Saipol coast population. And we think this is probably because of a dispersal route coming from the trans-Antarctic mountain area around. So like during times of open seaways in the past, for example, when this whole West Antarctic ice sheet had collapsed, or here maybe you can visualize what Antarctica looked like when this ice sheet had collapsed, and that actually it nicely illustrates how you can see these open seaways and the potential for a little rafting mite to catch a ride on the current to a new habitat like around the corner. And so now my last slide, coming back to how we think of Antarctica today as the single solid landmass, but when we understand those kind of past landscape evolution changes and how this has led to the present day genetic diversity, that's how we're using these DNA barcoding methods still in a really meaningful way. And with this, I would like to thank all of the organizations who have been involved, my current employer, because this is kind of like a side project that I'm trying to publish, and all of the previous people who were involved in the collections, and of course my keen co-authors who are helping me put together this paper, and I hope to share it with you soon. So you can follow me on Twitter to watch this, or maybe my email address will be available somewhere through the conference. So yeah, thanks very much for your attention.
Thank you very much, Gemma, for this talk. Very interesting. I don't know if there is some question in the chat or in our. No, perhaps Claudia.
Yeah, I have a question, Gemma. Very nice talk. Thank you. It caught my attention that you mentioned that the dispersal mechanism is by wind, and it's kind of a I don't know, like tricky to think that if they are dispersing all the time by wind, how you can find these very structured groups with a lot of speciation processes occurring to confirm all the species that you have found.
Yeah, so it's really good you pick up on this point, actually. And I mean, we're always thinking about the mechanism for dispersal when we're thinking of collembola and mites in Antarctica. And this was something from this original literature from like the 60s, where they were kind of writing down their observations and what they thought. And so I guess that's where the idea of wind dispersal has come about. And we've tried before with wind traps to try and actually catch springtails or mites or something in the air, and we never really found anything. But I left it in there because maybe it is possible, like over short distances. But I would say, yeah, it's and maybe for the mites, I'm thinking it might be more possible they're dispersed by wind because they're a little bit smaller and maybe less likely to be like completely dried out when they get put up into the air, whereas the collembola just desiccate straight away and they don't really survive this transfer. But I think in springtails they showed like maybe 30 centimeters or so it was possible. But yeah, if you're really thinking of this large spatial scale, then maybe it's more likely they are traveling by water and like floating or rafting on the top of the water. So yeah, or I mean, also they can walk or catch a ride on a bird or something like this. But yeah, really good question. And I wouldn't have any evidence to support that they are actually dispersing by wind. But
yeah, I think it's a kind of like an open debate about how are the or what are the mechanisms for terrestrial dispersal, because we are still finding a lot of the structure and the mechanism still doesn't like appear or have real proof of evidence that they are actually transported. So thank you.
Okay, so yeah, this reflects then what I was kind of feeling as well. So this is good. Thank you.
Thank you.
Thank you very much, Gemma. We are on time. Thank you for your talk. We will go further and smaller organisms. We are going to the next talk with Guillaume Schwab, who is in France, but actually presently working in Chile, and he will present us this talk exploring the microdiversity within marine bacterial taxa toward an integrated biogeography in the Southern Ocean. Oh, what's a nice title. So, Guillaume, you can share your presentation. I think all is okay. You can start.
Okay. Hi, Elie. Thanks for the introduction. My presentation is visible, yeah? Okay, so I will start. First of all, thank you for the opportunity to present my work. The aim of my talk today is all about convincing you that microbes do deserve to be included in the biogeographic atlas of the Southern Ocean, because their distributions are governed by the same ecological processes and the same oceanographic features as the microorganisms. So there are two emblematic and ecologically important oceanographic structures in the Southern Ocean: the Antarctic polar front that separates sub-Antarctic and Antarctic regions, and that is characterized by the strong thermocline. And at the other end, we have the Antarctic circumpolar current, which is the longest and the strongest current on Earth, and that flows clockwise all around the Antarctic continent. And both currents are known to shape the pelagic and Baltic fauna in two distinct ways. On the one end, we have the APF, the Antarctic polar front, that constrains the individual dispersion. For example, it had been evidence that this current drives the population differentiation of the Gen 2 penguins through genetic drift, leading to very high phylogeographic structure across the Southern Ocean. And for that reason, the Antarctic polar front is considered as a barrier between Antarctic and sub-Atlantic provinces. Contrastingly, the ACC promotes west to east long-distance dispersion, leading to population genetic admixture among the distinct population of the Southern Ocean. And we do have evidence, as here with marine snails of the genus Siphonaria, that show that there is a very low or almost absent phylogeographic structure, resulting for this connector role of the ACC between the sub-Antarctic provinces. So what's about the microbes? We have some evidence that the APF may act as a barrier. For instance, this nice paper from Rice and et al. Reported that there is actually a shift between the Southern Ocean and the South Pacific Ocean based on the whole bacterial community obtained from a 16S, MySeq sequencing. But there is several gaps that remain. First, they use a relatively narrow transect in the Southern Ocean. They look at exclusively the surface waters, and they do not look into the microevolutionary processes at populational level. And we do not have evidence of any connectivity through the ACC. So considering that the effects of these two currents are completely unknown on bacterial population, our objective was to unravel the genetic and phylogeographic structure and connectivity of marine bacterial taxa to evidence the role of the Antarctic polar front and the Antarctic circumpolar current. So to fulfill this objective, we needed the right model in the right ecological context. We know that working on similar habitats will minimize the importance of the environmental filtering and maximize our ability to detect neutral genetic variation that is associated with the biogeography. So for this reason, we used the sea urchin Labatus as a proxy of habitat homogeneity for our microbial biogeography study. This genus comprises various sibling species that are distributed in distinct provinces of the Southern Ocean. And all these species have roughly the same ecology and live in very similar habitat conditions. So we perform wide-scale sampling, including two locations in the Patagonia. Oops, sorry. With the species Abatus cavernosus, One location in maritime Antarctica with the species Abatus agassizi, and one locality in the Kerguelen Islands with Abatus cordatus. Then, as we know that the contribution of geography is stronger on habitat specialists, we decided to work on the gut tissue of Abatus, because we know from a previous study that the gut tissue of this species acts as a filter of the environmental surrounding bacterial community, filtering just a few taxa within the gut tissue. And among all these taxa, these few taxa, we choose the Spirochaeta genus because it was the most prevalent and abundant genus in the Abatus gut. And we suppose that it was very specialist from this kind of habitat. And we expect to maximize the detection of phylogeographic signal. So we started with a classic extraction of DNA, genomic DNA from our Abatus gut tissue, followed by the classical metabarcoding approach on the 16S gene. We filtered out the 10 most abundant operational taxonomic units that were affiliated to the Spirochaeta genus. And here is what we obtained in terms of their abundance and distribution across the sampling sites. And we already can see, just at the middle of the OTU, that we do have a non-random distribution of this OTU according to the sampling site. But for our phylogeographic study, we need to focus on a single O2 to get into a more populational level. And for that, we selected the most abundant O2 affiliated to Spirochaeta that was also present in all the 4 regions of our sampling. So we get all the sequences affiliated to this OTU7 of Spirochaeta, and we process them with the pipeline oligotyping that's allowed to discriminate the biologically relevant variability from the sequencing errors. And thus we can move from the OTU level to an oligotype level that can provide a more resolutive view of the phylogeographic pattern. So we obtain with this pipeline about 96 oligotypes, and here is a haplotype network of all of these oligotypes. We all can see that we have some of these oligotypes that were exclusively present in some location, while others were completely shared between among all the localities. And then we compared the genetic and phylogeographic differentiation of all the sites. And all the pairwise tests were significant. As expected, the 2 Patagonian sites were very similar in terms of genetic and frequencies of the oligotypes. And on the other end, we, as expected as well, the Patagonia and the West Antarctic Peninsula oligotypes were more dissimilar. Please note that, interestingly, there is the oligotypes from Patagonian site PAT1 are more similar to the Kerguelen site than with the Antarctic sites, suggesting that there is somehow a connectivity between these 2 localities. Then we tried to estimate the genetic flows among these distinct Spirochaeta population within the O27 from Patagonia, Antarctic Peninsula, and Kerguelen Islands using the Lamarck software. The flows were estimated through the number of effective migrants per generation among Spirochaeta population. And I show you here the mean values for obtaining with 10 runs and the n values above 1 were considered as relevant. So let's decompose this table. First, we confirmed the presence of a high genetic flow between the two populations of Patagonia that were pretty expected. Second, we detected a substantial gen flow from the Kerguelen Islands towards the two Patagonian sites. And we propose that these two flows follow the clockwise circulation of the SAC. We detected a low but significant flow coming from Kerguelen Islands towards the West Antarctic Peninsula location. And we do know, thanks to the literature, that that kind of gen flow are possible thanks to storms or strong winds or oceanic eddies that can occasionally disperse some particles from Kerguelen Islands toward the West Antarctic Peninsula. And finally, we do not detect any significant flows between between the West Antarctic Peninsula locality and the two Patagonian sites, suggesting that there is actually a barrier impeding the dispersion of Spiracata particles between these two provinces. So to sum it up, first we validate our methodology combining a 16S metabolite coding approach and the oligotyping pipeline to decipher the phylogeographic patterns of no culturable bacteria taxa at a populational level. Our study unifies the biogeography between the micro and the macro using comparable genetic resolution, the same tools, and depicting consistent phylogeographic patterns that are shaped by the same microevolutionary processes and the same oceanographic key structures across the southern oceans. All these results are published and accessible by scanning this QR code here. And with this, I'd like to thank the co-authors of this work for their precious collaboration, all the people with which we collaborate to obtain our samples and improve our bioinformatic pipelines, and as well all the institutions for the logistic, the material, and economical support they provide to us. And with that, thank you for your attention, and I will be pleased to take any question.
Thank you very much, Guillaume, for this talk. Very interesting to see how we could go inside the intraspecific diversity in bacteria as we do it for a long time for macrospecies. We have some time for question. There is one in the chat from Leah Ramirez. It asks if oligotype are similar to SIT Yotus.
What is a Zotu?
I don't know. Leah, if you want to...
It should be a unique sequence O2 or something like that. Yes.
It's, yeah.
It is quite similar at the exception that we have for the oligotypes an entropy analysis that allow to select the sites that could be interesting for discriminating the sequences we have.
No. Okay.
So, Guillaume, you spoke about to combine or to look at this bacteria in habitus and different part. It means the external sediments, gut contents, and tissue of the gut. Did you see some difference between them? Are you studying this part of this comparison?
We have actually a student that is working on that with a strong hypothesis that the phylogeographic structure we observed would be stronger when we are looking at bacteria that are associated to the gut tissue in comparison with the bacteria that are associated with the external sediment. For that reason that I said that habitat specialists are usually expected to have stronger phylogeographic patterns. So we will have a nice talk from Melanie in Tuesday, I think, or Friday, and we will know more about that hypothesis.
Okay, if you have the information, you can put it on the chat of the name of the session and what time more or less. Thank you very much,
Yes.
Guillaume.
Thank you.
We will move now from Europe to Americas with Elie Ramirez from Chile, that will present us the talk, a high degree of novelty and share metagenome assembled genomes between ice-free area impacted by penguins in Antarctic Peninsula. Elie, you can begin. It seems all is okay. You should, Elie, you should open your microphone.
Yeah. Hi. Well, I'm saying that I'm super excited to present my work. Thank you for the opportunity. And yeah, my work, my name is Elie Ramirez, and my work named High Degree of Novelty and Shared Metagenome Assembled Genomes, MAX, between ice-free areas impacted by penguins in Peninsula Antarctica. This work was done in collaboration with BASE, CATG, and Max Planck Institute for Marine Microbiology. So here I want to show you a picture of, as an introduction, as an ice-free area in Peninsula Antarctica impacted by penguins. We can see here how animals can impact the land, mainly by their depositions, altering edaphic factors, and also soil microbial communities. In our previous work that we can see in the publications, we study ice-free areas impacted by different marine animals, and we conclude that penguins reduce soil microbial diversity and increase metabolic pathways as denitrification in these soils. So in this study, we want to know the impact of different species of this penguin, Pygoscelis genus, P. antarcticus, P. adelie, and P. ginto, in the composition and distribution of soil microbial communities in ice-free areas in Antarctica. These three species are distributed through the Antarctic Peninsula and has been reported that possess a species-specific gut microbiome. So we are interested to know if we found difference in the soil that they settle. To do this, we sampling 18 ice-free areas along Antarctic Peninsula. Here we can see the map of the different sampling sites from Pygoscelis, Adelia, and Antarcticus. We take samples from triplicates. So to assess the microbial diversity, we took the top surface of the soil of each sampling site and storage for following analysis. Then we performed the DNA extraction and performed shotgun sequencing using Illumina platform. The assembly, we made assembly at the beginning of the MAX. MAX are metagenome assembled genomes. And after that, we do the analysis. First, we quantify sequence diversity of each metagenome using non-parallel diversity index. Then we perform taxonomy annotation using GTDB database. And also we question about the novelty of the MAX that we recover. We determine the MAX distribution according to each Pygoscelis species. And finally, we perform community network analysis. So this is our results. We found, we obtained 54 metagenomes representing the 18 ice-free areas in impacted soils by Pygoscelis species. We've been more than 1,000 of MAX obtained by the co-assembly by site. And also we obtained more than almost the half were non-redundant MAX. So here from the results, we can see the metagenomic data set. We evaluate the sequence diversity using non-parallel index, and we can see that Adelia species present significantly lower sequence diversity than chinstrap and ginto species. Then we evaluate the taxonomy diversity at phylum level, and we can find that Protobacteria and Actinobacteria were the most abundant max in our non-redundant max compendium. And we are able to recover Archaea max 6 specifically. Also, we want to know the novelty of the max that we obtain. And here we can see this was based in the GTBB database. And we found only 2 of our non-redundant max recover can be assigned as a known species in the database. So we can conclude that more than 99 of the max likely represent a novel species not included in the database. Also here, we do recruitment rate plots. So we plot the percentage of the recruited reef recovered from soil impacted by Adelia, chinstrap, and ginto penguins. And we can see that beans obtained from Adelia, for example, recruit reefs from Adelia, chinstrap, and ginto metagenomes. And also this happened in chinstrap and in the ginto animals. So we can see that the community, the microbial community here are shared between the sites and between the penguin species. Also, we perform network analysis with the 1009 max that we obtained. And here we graph the connection between the max, consider 99% of any, that it means that belong to the same species. So here we can see that the circles are max and the color are related to the site that we obtained. So here we can see that many clusters that indicate that max are present in different sampling sites, and these sampling sites belong to different species of penguins. So also this confirm what we see before, that max are shared between the sampling site and the different species of Pygocelis. So in conclusion, we assembled more than 1,000 max, and the compendium was approximately 173 non-redundant max, and things there are Archaea. More than 99 of the max likely represent a novel species not included in the database. Also, we see that ginto and chinstrap impacted sequence presented significantly higher sequence diversity than Adelia impacted soils. We see a high degree of similarity of max between the ice-free areas. And this, in total, we can conclude that high degree of novelty and shared microbial communities between sites suggests similar microbial communities between these ice-free areas that we sampled in Peninsula Antarctica. So that it's all the results. I want to acknowledge to base from the support, and also other institutions such as CTIT and Max Planck Institute for the help. Thank you so much.
Thank you very much, Leah. We have time for a question.
Yes.
Quite a time. So if there are some first microbiologists in the room, waiting for that, I would like to ask you, Leah, what can generate the difference in the different colony penguins? I think Adelie was different from the other one. What could be the reason or the kind of hypothesis for that?
Well, I mentioned that there are publications that shows that gut microbiome in these different species are species specific. So this could be for the what they feeding. I don't know. maybe some of them are feeding. I know they feed krill and fish, so probably the proportion of this can be having an impact in the gut microbiome, for example. But that is what it was important for us to know if this differential gut microbiome could be an impact in the soil also, in the taxonomy of the microbes that are in the site where they settle.
Okay, it was also, I think, the question that asked Leah Cabrol, who asked if you can relate the difference between penguin species to any environmental characteristic. There are some important difference between the breeding colony sites where the penguins choose for reproducing, like more snow or.
I read some that they have difference in the breeding. I'm not sure if there are. I think all of them doing in the land and not in the ice, but I know that there are differences in the timing that they do. But yeah, I think it could be very interesting to talk with the penguin people that know more about the behavior of the penguin to complement this work, probably. Yeah.
Okay, there is another question. I don't know if you can look to the question answer section, because
okay,
to understand the question.
Oh, yes. They asked me about the throughput of the completeness and contamination to select the max analysis. Yes, we do this, and we base it on Bauer's paper that they categorize the max from low, medium, and high quality for the contamination and completeness percentage. So we work with the medium and high max that we obtain. So this compendium that we obtain are based on this medium and high max. So yeah, but we checked that. And Leah asked, could you relate the difference between penguin species to any environmental characteristics? Oh yeah, that's the question that you made.
Okay, if there is no more questions, thank you very much, Leah, for this very interesting talk and this amount of work we have shown. And we will stay in Chile with a pre-recorded talk of Constanza Millán. I don't know if SCAR team is already here to initiate this presentation. Seems not. Oh, well, this is, well, just two pre-recorded, no free. SCAR, are you here?
Yes.
Okay, we'll wait 2 minutes to see if
We have like 3 minutes.
here. If this time there's some comments or questions, I see that Melanie put information about her talk on Friday.
Mm-hmm. Yeah, I put it on the chat also. The microbiologists are talking.
Yeah,
in the chat.
Guillaume asked a question, I think for Leah. Leah, are you still there?
Yeah, I answer in the chat.
We can also say hi to Pete Convey, who couldn't be here today because he's in Svalbard in the Arctic, so it doesn't have very good connection to actually connect today. So he was one of the conveners. So thanks and hi to him. And Scar is
not really here. So perhaps we could move the next talk and hope they will appear at some time to see the presentation of Constanza.
It is expected to begin at 11:30.
So I think we'll move and that doesn't work very good.
Yeah, okay. Should I share the presentation? Yes, yes, please.
Okay.
Okay.
Okay, it will start.
Constanza Millán, please.
So we'll have the talk of Constanza Millán about rafting and genetic structure of Siphonaria limpet in the Southern Ocean. Also from and from base.
Yes.
So Scar, we are ready. No, it's a response. I said they will.
No, it looks like it was sharing something. But, okay. There you go.
Hi, good afternoon, everyone. My name is Constanza. Because I'm doing field work, but I've left this recording.
Is it like that in all the sessions? Have you seen it before? Okay, it seems here we are.
Hi, good afternoon, everyone. My name is Constanza Millán Medina, and I'm so sorry I couldn't be here today virtually because I'm doing field work, but I've left this recording of this presentation titled Rafting and Contemporary Genetic Structure, Revisiting Siphonaria naturalis in the Southern Ocean, which was part of my master thesis. So we know that dispersal, either active or passive, is a fundamental aspect for the dynamic of marine species, their distribution and connectivity. The Southern Ocean has multiple oceanic fronts acting as barriers, and yet there's plenty of evidence that there is much more cross-frontal exchange than we previously thought. Benthic and intertidal invertebrates, even those that lag a dispersive stage, such as the pelagic larvae, are known to cross fronts and disperse across the Antarctic circumpolar current through surface rafting, in which they hitch rides on organic or inorganic materials such as microalgae that eventually drift across fronts and navigate currents. But how do we know they cross fronts? Well, there's plenty of studies, genetic studies in the Southern Ocean, in which species distributed along various subantarctic islands show high degrees of genetic connectivity between long distance, displaying this classical star-shaped haplotype network, where at least one haplotype is shared between almost all sample populations. Moreover, the rafting and macroalgae by excellence in this area, the real Antarctica, also fits this connectivity pattern, and most importantly, has a single widespread haplotype not only through all sub-Antarctic islands, but also through southern South America as well. However, given that connectivity is such a relevant feature in the dynamic of marine species, moreover in the Southern Ocean, it also raises a question. Are these patterns ghosts of past genetic connectivity, or is this an ongoing process that maintains ecological genetic connectivity? Hence, the title of this presentation, Revisiting Cyphera naturalis, which is a gastropod broadly distributed through Patagonia and sub-Antarctic islands, yet has limited dispersal on its own. A previous study by Gonzalez-Weber and colleagues in 2018 revealed that along its wide distributional range, this species was a genetically cohesive species and yet was structured in three groups: Falklands Islands, South Georgia, and a widespread cohesive genetic group made up of South America, Kerguelen, and Macquarie Islands. Such patterns then demonstrate a high genetic connectivity by rafting like the examples I mentioned before. So the aim of this study was to further explore the genetic diversity and genetic structure of Cyphera naturalis to determine if there was any contemporary gene flow amongst its wide distribution in the southern hemisphere. And for that we use a genomic approach with SNPs because the molecular tool we use matters. We sampled 8 geographical sites, 3 from Patagonia, 1 from Falkland Islands, 1 from South Georgia, and 3 from Kerguelen Islands, and we retained 116 individuals and 5,500 SNPs approx. After trimming and cleaning our dataset, and with this dataset, we estimated classical genetic diversity statistics, performed different genetic structure analysis, and determined gene flow through different approaches. As you can see in this table, across all sampling sites, various diversity indexes were extremely low, with the exception of Falklands, which had a higher allelic richness, higher amount of polymorphic loci, etc. If we group, however, these 8 sites into their 4 major geographical areas, Patagonia, Falklands, South Georgia, and Kerguelen, we can see that Falklands still maintains its high diversity and more than triples the private alleles of the next diverse area, which is Patagonia. We can also see that South Georgia displays a high percentage of polymorphic loci. However, it has scarce private alleles, meaning less private diversity on its own. The genetic structure analysis of these 8 sites consistently show 4 genetic groups in agreement with these 4 major geographical zones: Patagonia, Falklands, South Georgia, and Kerguelen Islands. However, Patagonia and Falklands are markedly differentiated in purple and orange in this graph and all the graphs. However, South Georgia and Kerguelen Islands are not as markedly differentiated in blue and green. Regarding connectivity analysis, such as IMS, which illustrate mean migration rates, we can see that it represents 2 important things. There's quite an orange or even brown halo around Falklands and Patagonia to a lesser extent, which signal the presence of barriers to gene flow. And on the eastern side of this map, there's a blue tint around South Georgia and Kerguelen Island, which is a signal of migration between these 2 populations. Gene flow estimates also show a higher exchange rate between these 2 populations, and not significant between the others. Different statistical approaches help us clarify that this exchange is slightly higher from Kerguelen to South Georgia than the other way around, which is also easily illustrated by these Kerguelen blue bars in the structure analysis that are within the South Georgia green block of the population. However, when we also consider the self-recruitment dimension, which is the diagonal here, we can see that most of the individuals stay within their population of origin and do not migrate effectively. So how can we make sense of these patterns and how can we explain them? Well, the evolutionary history of Cyphera naturalis seems to be one in which rafting permitted the recolonization of the Southern Ocean, as discussed by Gonzalez-Weber and colleagues in 2018, yet nowadays this rafting event seems to be more likely occasional and don't support a widespread gene flow like the center haplotype suggested, spanning Patagonia, Kerguelen, and Macquarie. Instead, there's different ecological features that have promoted the genetic differentiation of these 4 groups nowadays. On one hand, we have the Southern South America scenario, in which Patagonia and Falklands are quite geographically close. They're almost 500 kilometers apart, and yet are quite highly differentiated as a byproduct of glacial cycles. We know that the Pacific coast was almost covered by the Patagonian ice sheet, and the Atlantic coast receded and exposed the continental slab, all which extinguished local biota. Meanwhile, the Falkland Islands were less affected, constituting most likely an Ice Age refuge supported by genetic and geological analysis. The marked genetic differences between these nearby areas are nowadays sustained most likely by different biotic and abiotic conditions such as temperature, salinity, oxygen concentration, and most importantly, the so-called Malvinas current, in which surface drifters analysis can, as you can see, there's quite a few particles that flow from the Pacific coast, yet they don't show any incursions, or at least not frequent incursions, to the Falkland Island, effectively isolating because this arm of the current goes around the island but does not penetrate the ocean between. On the other hand, Drupal Antarctica, this rafting vehicle, so to speak, not only does it have a single shared haplotype between Patagonia, Falklands, and other sub-Antarctic islands, but recent SNP analysis also distinguished a single genetic unit comprised of all of these areas, here in blue represented, and also particle models illustrate all these pathways or highways, so to speak, that Drupal Antarctica can traverse. However, Siphonia naturalis, its passenger, does not, exhibiting quite a restricted genetic connectivity and only between islands within the circumpolar Antarctic current. Interestingly enough, the CAC flows west to east clockwise, and there's gene flow signals from South Georgia to Kerguelen. However, our data supports a higher magnitude of gene flow from Kerguelen to South Georgia, which is still clockwise, but it's a much longer journey, almost 16,000 kilometers of oceanic rafting, which is plausible, but most likely stochastic, low frequency, and nowhere near the purported high connectivity that phylogeographic genetic studies suggest, which emphasize that the choice of molecular marker is highly important to ascertain the levels of past and present connectivity within the Southern Ocean, because while rafting may be an inequivocally effective dispersal method for macroalgae, its passengers dispersal is most likely up to a variety of conditions and therefore must be carefully regarded. So that's about it. If you got any questions, comments, observations, please don't hesitate to write me at the email that's on this final slide. Thank you for the attention, and thank you all of these organizations for the funding, because without them, the development of this investigation could not have been possible. Thank you.
Okay, this was a talk of Constanza Millán. If there are some questions, I could try to answer as I wrote co-author of this work. Some question about rafting and effectivity of this dispersal mode. No. Okay, we'll let the email of Constanza in the chat if someone want to ask something directly, and we will move to the next talk. Moving to North America, we'll have the talk of Kevin Billick. There is there and back again, what we can learn about the resilience of Antarctica. Oh, there is problem, the title, cryonototenoid fishes from their gain and losses following past climate. So we go back to the fish. Kevin?
Yes.
Okay, if you can share your presentation. You would see it's working good. Okay, you can start.
Greetings, everyone. So as mentioned, my name is Kevin Billick, and for those of you who don't know me, I'm interested in understanding how physiological systems have been shaped by evolutionary history. And I've spent a lot of my career trying to look at these heat responding pathways and heat tolerance in cooler fishes to see how they've suffered attrition as these fishes have evolved in colder environments and what that means for their ability to deal with the changing climate. Thanks to some nudges by Scott Santagata, who had a number of very fun workshops on molecular evolution, tried to take a bit of a broader view and genomic focused view in terms of how evolution in the cold is shaping a lot of these biological systems in these fishes that are adapting both the cryonotothenioids as well as a number of Arctic lineages as well. Focusing today on the cryonotothenioids though, I used this molecular evolution approach to explore how evolution in the chronic cold of the Southern Ocean has shaped a number of these biological systems over three major physiological transitions that we see in the cryonotothenioids. First, to introduce the physical environment, and this is generally something that I normally don't need to do at SCAR, but turns up a lot in other conferences. Antarctica is of course cold, but it is also a very cold, stable environment. And this should represent two separate evolutionary events. First, trying to deal with the extreme cold of this climate. And I bring that up because living in the Northeast of the United States, we have lots of really cold adapted fishes that turn up here in the winter. We have lots of fishes that are really good at dealing with the cold. I can point to the example of something like the winter flounder, which is characterized by a lot of antifreeze proteins and other important cold adaptations, but still survives water temperatures that go up into mid-20 C during the summer and are doing just fine. The chronic colds, that loss of thermal variability that we see in the Southern Ocean should have been, or would have been, a really important evolutionary event on its own. Something that I think of when I look at some of those systems that I'm particularly interested in, the heat shock response and heat tolerance, which would have suffered a lot of their attrition just on account of that chronic low temperature. And that second event, that chronic colds, would have been a source of relaxation of selective pressure that would have allowed genetic drift to occur, and is something that I hadn't been seeing a lot in a number of those prior genomic investigations of the clade, at least until Jacob Dane started to investigate a lot of them relatively recently, the last few years. So as a starting point, I was interested in terms of how these two things, this extreme cold and this loss of thermal variability, could have shaped those biological systems of the cryonotothenioids. What we're seeing from that positive selective pressure from the cold environment and what we're seeing from that relaxation of that chronic cold environment. And the hope was that this would have shown a lot of interesting things in terms of those stress responding pathways that I was interested in. But of course, I was very naive at the start of this little investigation. The study that we were looking at, myself, Xin Zhang, and Chiara Papetti, and later on Santiago Solas, tried to focus on these three major physiological transitions that we targeted as being particularly interesting to us. First was what happened with this transition to freezing temperatures, and we focused on the red-blooded cryonotothenioids, so those that had not yet lost the hemoproteins, to see if we were seeing any signature of adaptive change that was happening due to that onset of cold temperatures and freezing temperatures. Second, we wanted to look at this transition to the loss of hemoproteins. So the Antarctic fishes are one of the few vertebrates, and in fact, the only vertebrate as adults, where you have fishes surviving without the hemoprotein hemoglobin, and in some cases without myoglobin as well. This is a major physiological difference from that vertebrate mainstream, and something that we would have thought would have presented a major shift in selective pressure that we would see in the genes that we were looking at. And finally, and something to which I don't have very robust data yet, only the starting points of an investigation, but a number of these cryonotothenioid lineages have made the secondary transition back to temperate waters. And we're interested in trying to understand if these have shown any consistent change that has come with going from being in a chronic cold environment to back to a more thermally variable one that would tell us something about that ability of the cryonotothenioids to adapt to future warming. So to carry this out, again, it started out both with that conference that Scott Santagata put together, where I met Chiara Papetti, and later on roped my colleague from the Chris Cheng and Kirk DeVries lab, Shen Zhang, into helping with, to try and ask what biological systems were being shaped by that transition to chronic cold and that loss of hemoproteins. What could we learn from the existing genomic data sets that were out there to try and explore how these changes occurred, or what changes had occurred? And as I brought up on this last slide, this is something that we weren't attempting to do fresh sequencing on, but to make use of the existing genomic and transcriptomic data sets that were around. There's been a large transfer of new genomes coming out, but at the time at which we had launched this, there was a relatively modest set of transcriptomes for a little more than a dozen species, and genomes for about 3 or 4 of varying quality. So we targeted this transcriptomic resource that was out there because it seemed to cover a wider range of tissues that were available and was often provided a more robust data set to use. But it led to the first problem, which is that every single investigator describes their work as building a reference transcriptome. What that reference transcriptome is made up of varied greatly from one study to the next. So we focused in on those species that had a relatively large number, at least 4 tissues represented in their transcriptome, reassembled all of these using a consistent approach, and identified, searched them to make sure that they maintained a relatively stable level of quality compared to what we were seeing in the model species that we were grabbing off of Ensemble. We carried out ortholog detection to put our tests on a consistent set of contigs across all of these species, and then used the evolutionary hypothesis testing tool HiFi, suite of tools HiFi, to look at what genes were coming under either positive selective pressure or relaxed selective pressure. Ultimately, we had about 4,000 contigs across our suite of comparisons, or 4,000 putative genes with which to investigate what changes had occurred. In terms of broad trends, what we had found is that signature of relaxation of selection, either that the genes were showing a loss of purifying selective pressure or that positive pressure was being reduced, was roughly equivalent or even larger relative to the usefulness of these tools than what we were seeing in terms of those positive selective pressures. Looking at positive selective pressure itself, we had found signatures of a number of biological changes that were appreciated already in terms of what had been investigated in the cryonotice units. First, that, to put it broadly, building things in the cold is hard. There was this general signature that biosynthetic processes was coming under positive selective pressure, but also that there were these more focused signatures of change related to microtubule assembly, something that Bill Chetrick's work has appreciated for a long time, as well as cell adhesion, a set of changes that we've seen a lot in terms of cold adapted organisms. In addition, we saw this set of genes related to cell cycle control coming under positive selective pressure, possibly on account of the fact that these fishes need to compensate for the rate limiting effects of cold temperature in terms of tissue growth and organelle growth as a whole. In terms of relaxed selective pressure, again, this is where I was originally hoping to see widespread change in terms of all of my heat responding pathways. Instead, what really stood out in terms of this set of genes is how much I was seeing the reductive traits that have come to dominate that ecological diversification of the clade. Genes related to craniofacial development, where we see a lot of that change in head shape in the group, as well as genes related to bone and cartilage development, something again that Jacob Dane has investigated a lot of, but that's also correlated with that change in terms of neutral buoyancy within the diversification of the group. One thing that was novel was the fact that we were seeing these genes related to DNA damage. Again, something that might be tied to that slowing of cell cycle due to the colder water temperatures might be related to the fact that there was less of a need to undertake DNA replication quickly. Key findings: clear signature of change due to both positive and relaxed relaxation of selective pressure. Positive selective pressure seemed to be shaped heavily by the fact that building stuff in the cold is hard, and relaxation of selective pressure heavily dominated by those reductive traits. Because time is running short, I will fly through these last few parts. But in contrast to what we were seeing in terms of that, in terms of the red-blooded notothenioids, the ice fishes showed a relatively small number of genes coming under positive or relaxed selective pressure relative to the red-blooded fishes. And in fact, to the extent that we were seeing any coherence, signature of selective change was positive selection and related to heart development. But again, this was a much smaller and much weaker signature than what we were seeing in the positive selective and relaxed selective pressure of those fishes as they were adapting to the colds. Something that was also seen in a number of these individual genome studies that were looking at this group. In terms of the Antarctic escapees, I will say we have only just started to look at this, and it is very much thanks to data sets that came from Santiago Ceballos to look at this. But we are seeing, at least from an initial screen, that there are substantial differences in terms of how many genes are coming under change selective pressure from the cold cryonotothenioids to these Antarctic escapees. And in fact, these are showing widespread differences in terms of which set of genes are coming under change selective pressure with each species. So this, for me at least, is undergraduate driven research. So it's moving in fits and starts and relatively slowly, but makes fun progress. I'm always excited to see every time I'm in the lab. And there is that finding that there's this major difference in terms of each of these individual groups in terms of what genes are coming under selective pressure. So wrapping up, I would just like to thank a couple of people, Scott Santagata, and again, for putting this workshop together that kickstarted this. I believe he is in the audience today, so thank you, Scott. My really awesome collaborators and people I will strongly suggest that anyone in the audience think about working with as well: Chiara Pipetti, Chen Chang, and Santiago. The undergraduate researchers who have helped a lot with this work as I've moved through it, living the Pui life here at Montclair. And the funding, a little bit of which has come from Montclair State University, my academic home, which has helped make this possible. Okay, any questions? And I will admit my question slide is entirely at the wrong pole. It is in the Arctic rather than the Antarctic.
Thank you very much, Kevin. We have time for some questions.
Yes, 2 minutes.
Yeah, like 2 minutes, some questions. I always some question. And I also work on some of these fish, in particular at Pacific. So I wanted to ask you what happened to the antifreeze gene. It still functions, and the species that went back to sub-Antarctic areas. It means
So,
there's still this amplification is still working.
So they seem to be greatly reduced in terms of their activity. There is also a quality genome paper coming out from Angel Riviera Poulin, who is working on a joint project with Chris Cheng and Julian Katchen. The preprint was about to go live, and unfortunately it's been delayed by a couple of days, but they're looking at Camphocephalus esox, and the antifreeze loci is a key area of concern because Chris Cheng is involved in that project. So of course she looked at it. I don't want to spoil anything because again, it is their study and I was just a very small part of it. But if you look for that, hopefully in the upcoming weeks, it will answer some questions in terms of the genetic change in the loci for these secondarily temperate species.
Okay, and about Pacific, most of the species are described out of Antarctic. There is a single species of Pacific among the 12 described. And it's only located in the West Peninsula. It means is it really an Antarctic genus?
Sorry, can you repeat the question?
Is it really an Antarctic genus? When you see the distribution of Pacific in Antarctic, it's just located along the West Peninsula. You won't find it like a circumantarctic species, and you will find in all the different sub-Antarctic islands in Patagonia and quite north.
What I would argue is in terms of how how cold it experiences. If it's under that extreme cold and chronic cold for at least a substantial part of its life cycle, that's something that I would expect is driving a cold adaptation that would make it an Antarctic species. But again, that's really going to be driven by what type of modality of cold it experiences.
There's a question there.
There is a question. Yeah, if perhaps Kevin, we have no time, but you can answer directly in the answer question section of the website.
Okay.
Thank you
happily.
very much for
Who was the questioner? I could reach out to them directly too.
What? Pardon?
Oh, who had asked the question, because I can reach out to them directly.
Santaga. Scott
Oh yeah,
Santaga.
yeah.
You can,
Okay.
okay, thank you very much, Kevin. If you can put out the presentation for the next speaker.
Mhm.
Thank you. So the next speaker is Sebastian Rosenfeld from Chile that will present us unveiling the systematic and taxonomy of the Antarctic and subantarctic gastropod Leviditorina caliginosa based on morphological and molecular evidence.
Hi, everyone.
Hi, Sebastian, you can
See, you can see the PowerPoint.
Yes, try to put it in presentation mode. Okay, you can.
Yeah. Okay. Hi, everyone. I will present this research entitled Unveiling the Systematic and Taxonomy of the Antarctic and Subantarctic Gastropod Leviditorina caliginosa based on morphological and molecular evidence. Okay, the genus Leviditorina is one of the most common and abundant groups that inhabit the shallow coast of Antarctic and subantarctic environment. Also, you can find several species of Leviditorina across the Southern Ocean. The species of the genus Leviditorina inhabit, oh, sorry, I have problem with, oops. I see. The species of the genus Leviditorina inhabit shallow habitat and can be associated with macroalgae. One of the characteristics of the genus is that it has 21 recognized species and that can be found across the southern hemisphere. Another characteristic of the genus is that it has a life cycle with direct development. That means that the female put the egg masses on a rock or on a macroalgae front and then hatches a juvenile. Commonly, this type of development is related with a narrower distribution for this species. And if you can see, mostly of the 21 species of Leviditorina present this type of distribution, except for Leviditorina caliginosa, which is the exception from the genus. Nevertheless, with the last manuscript that we published recently, we found that this wide distributed species, Leviditorina caliginosa, in fact represent 7 different lineages across this distribution. So we have many doubts about the taxonomic status of this different lineage, and this is one of the objectives of this presentation. To do this work, we collected a lot of material across the Southern Ocean and many subantarctic islands from the intertidal and subtidal zone. So we did 2 analyses, a morphological analysis. We review all the taxonomic literature for the group, and we describe the regular morphology of each lineage. And also we did molecular analysis using 2 mitochondrial markers. The multi-locus phylogeny recover the 7 lineage when a strong statistical support in each node. This is a picture with the several lineage, and for our own nomenclature, we put correlative numbers in each different lineage. We put lineage 1, 2, and 3 from the Strait of Magellan new species, the lineage 4, which is present in South Georgia Island, Crozet, Mayon, and Kerguelen Island, and Antarctic Peninsula, and lineage 5, 6, and 7, which are present in the southern part of the Magellan region and also in the Falkland Malvinas Island. So we start with lineage 4. When we review the literature from the South Georgia Island, we found that Martens and Pfeffer in 1886, they describe a species called Leberitoina venusta, that it has a very similar morphology with Leberitoina caliginosa, except for one criteria that correspond at this white columellar callus, white expanded columellar callus toward the umbilicus of the shell. So this was the criteria that Martin used to describe this species. So when we analyze the lasted material that we have from the South Georgia, we found some individual with this morphology. So we finally can identify morphologically Leberitoina venusta. Unfortunately, in the last manuscript, we can use this data in the phylogenetic analysis. But when we reanalyze all the sequence with the new material, we found that all the Leberitoina venusta individuals integrate a strong clade with the other individuals from the lineage 4. Additionally, when we analyze the radular morphology of Leberitoina venusta, we found that the morphology of the different teeth in the radula are identical to the morphology that Arnaud and Bandel described from population of Leberitoina caliginosa came from Kerguelen Island. So with this evidence and with the priority of name, all the lineage from the lineage 4 correspond in fact to Leberitoina venusta. When we analyzed the phylogenetic analysis, we found this lineage that was restricted to the Falkland Malvinas Island. And when we reviewed the literature, we found that in the Falkland Malvinas Island, there was a species described by Preston called Leberitoina latior. And when we match the morphological characteristic of latior with our material for the Falkland Malvinas Island, we can confirm and validate this species. We can validate taxonomically this species. Also, in the manuscript that is in development, we are going to show the first description of the radular morphology of Leberitoina latior. The case of Leberitoina caliginosa was very complex to resolve because when we reviewed the original description made by Gould in 1849, we found that the description was very general, and also the description of the type locality was very general. He put Tierra del Fuego, or Feuerland, and the problem is that Tierra del Fuego is one of the biggest islands in the Magellan region. So it has a side coast to the Atlantic Ocean, toward the Strait of Magellan, toward the Pacific Ocean, and also the Beagle Channel. So in which part of this big island is the type locality of Leberitoina caliginosa? Also, with our molecular analysis, we know that we have a lineage that we found in the Strait of Magellan part of the Tierra del Fuego. So the scenario was very complex. Fortunately, we found the narrative of the United States exploring expedition where the material of Leberitoina caliginosa was collected. And in the chapter 6 of the book, that is called Tierra del Fuego, same as Gould used it, the Captain Wilkins made a very specific description of the base camp where the ship was, and that place was Orange Harbor or Orange Bay in Hoste Island, an island that is disconnected from Tierra del Fuego. So the type locality of Leberitoina caliginosa will correspond to Hoste Island and not Tierra del Fuego. So with this evidence, together with the morphological and molecular data, we were able to designate all the population present in this geographical area and the rest of the population that integrate this clade as Leberitoina caliginosa. And that includes population from the Navarino Island, Hoste Island, Horn Archipelago, this white morphotype that we found only in Horn Island, and this morphotype that we found in South Georgia. So with that in mind, which are the new species? So the new species of Leberitoina are the lineage 1, 2, and 3 that are present in the Strait of Magellan, and the lineage 6 that is present in Horn Island. If you can see, the different lineage not only present molecular difference, but also present external morphological difference in the shell, but also we detected a difference rabular morphology between the lineage, mostly present of little denticulate at the end of some cusp in the rachidian tooth and in the lateral tooth. So what is the new scenario in Magallanes with this result? In Magallanes, in the Magallanes province, we have now 3 new species restricted at the moment from the Strait of Magellan. We have Leberitoina caliginosa, which distribute in the southern part of the region, and also in the Horn Archipelago, and in the Falkland Malvinas Island. We have Leberitoina latior restricted to the Falkland Malvinas Island, and we have a new species also at the moment restricted to the Horn Archipelago. From the Antarctic and sub-Antarctic environment, the new scenario is that we have Leberitoina caliginosa who reach the South Georgian island. We have this wide distributed species, Leberitoina venusta, which is present in South Georgia, the sub-Antarctic island Marion, Crozet, and Kerguelen, and the Antarctic Peninsula. And the problem that we have with Leberitoina umbilicata, like Claudio mentioned it yesterday in the session, when we identified the species umbilicata, Leberitoina antarctica claviformis from individuals from Antarctic Peninsula, we found that genetically they integrate the same clade. So we thought that maybe it's the same species, but we don't have all the material from the type locality of this species. So this case needs to be further analysis with more material. And I want to thank all the student national and international collaborators who helped me in the development of this project, also the Millennium Institute of Biodiversity of Antarctic and Soledad Ecosystem, and the regular Fondesit project led by Dr. Claudio Gonzalez. And thank you very much.
Thank you very much, Sebastian.
Yes,
we have some question. Maggie asks you, do you have any data correlating the morphotypes and genetic with diet and or substrate on which specimen collected?
No, at the moment, we don't have any data about the diet of any of these species. Only the molecular markers and the morphology.
Yeah, Maggie, why you were asking that, or thinking about like ecological speciation, or you can talk to Maggie if you want. Maggie? No? Okay. The ecological ramification. Okay. I have a question also in this sense. It's very intriguing that one of these clade show this extended distribution, while the author can show very restricted distribution. What could be the difference between these species? They are very similar in form and size and ecology. That may explain that such difference in distribution range for this species in particular.
Yeah, we saw that we need to develop more evidence about some habitats of this new lineage. For example, because the lineage that we have in Horn Island, the new species is a very small one, black one, and we found in the intertidal zone, but in a place with dead barnacles inside the valve of dead barnacles. And the Lebrituina caliginosa that is present in Navarino Island inhabit the high intertidal zone. Maybe the lineage 4, which is in South Georgia, is more related to macroalgae. And maybe it's more related to, I don't know, Durbilia or macrocystis, and maybe by rafting, they can reach this wide distribution. Is a possibility. But in the Magallanes region, I have photos that the species are not related to macroalgae, for example. Only some parts of the intertidal zone, and the lineage from Horn Island is very specific. That is my idea, but we need to test that. We need to more sampling effort.
Okay, Sebastián. There's some other question. If not, we will move to the next speaker. Jacob then will present Déjà Vu, exploring replicate radiation of perciform fishes to understand the genetic and development origins of key threats. Back to the fishes. Jacob. Nathan is not here. It's in the systems. No, it seems that Jacob is not here. I don't know if Scar is there. We didn't have information of a pre-recorded talk for Jacob.
So we don't have any pre-record video of Jacob.
No.
And here it's maybe we recorded. It's not Jacob, it's Jake. I'm sorry about that.
Yeah, we're a bit confused about Jake or Jacob, ma'am, because the name is the same.
Okay, sorry. So, yes, please, can you share your screen? Are you having presented?
Actually,
it's Jake Dane.
Oh, okay. Jake.
Jake Dane, yes. Thank you.
Yes, I'm here. Do you hear me?
Yes.
Yes, you were
okay, hold on.
Not in the pan section. Okay, now you should.
All right, I think we're cooking now. Let me see here. Where is sharing screen? Do I have permission to do that? I don't see that I have permission.
Yes, I think so. Because you are in the.
Yes, you are set as panelist. Maybe a Scar need to give you more credentials to share.
Oh, there. Okay, excellent.
Okay.
All right, is that good? Excellent. All right. Sorry about that. So today I'm excited to talk about two distantly related fish clades, one in the marine environment around Antarctica and the other in a freshwater lake in Siberia that have undergone what I think are very, very similar adaptive radiations that form something of natural biological replicates that might be helpful in understanding universal principles of adaptive radiation and trait evolution. So this plot is looking at lineage diversification rate among acanthophore fishes, which are about 2/3 of fish species and actually about 1/3 of all vertebrate species. And these two red bars here indicate lineages that have adapted in high latitude environments. And so what I hope you can appreciate is amongst all of this diversity, there's one clade in particular that just dominates the species richness at high latitudes, and that is the perciform fishes. This group includes Antarctic notothenioid fishes, as well as eelpouts and snailfishes, which are bipolar, and also can be found in extreme deep seas. There's other fish as well, rockfish, darters, stickleback, groupers as well fall into this clade. And so while they stand out as having repeatedly adapted into these similar and often very extreme environments, they're actually very distantly related even from one another, as you can probably appreciate from these photos. Last sharing a common ancestor probably about 100 million years ago or so. And so the hope is that we can maybe treat these radiations as something of biological replicates to ask if there's something, some sort of common genetic signature underlying their adaptive radiation, and if there's some shared genetic potential for these fish to diversify into these particularly extreme environments of polar and deep sea environments. So today I'm going to talk mostly about the sculpins in Lake Baikal with some comparing and contrasting to what we've seen in Antarctic notothenioid fishes. And these radiations are very, very similar in that they both have occurred at high latitude. Both of these are occurring at very cold temperatures. In both radiations, you have this rapid endemic radiation of a population that is responding to environmental change. In the case of Antarctica, it was the cooling of the Southern Ocean, and in Lake Baikal, it was the expansion of a rift lake and deepening of the rift lake. In both cases, these families represent the dominant fish biomass in their respective environment. And somewhat critically, diversification of both clades has occurred along a depth axis, both into deep sea or deep lake-like environments, as well as into the water column through evolution of pelagic characters. And in both cases, they've done this in very similar ways, which I'll get into in a minute. So this is just a picture of the Baikal sculpins to demonstrate some of their incredible diversity. What we can appreciate is that not only has this rapid radiation generated a variety of forms, including increased body size, differences in craniofacial shape, and trophic patterns, as well as reduction in eye size, changes in fin proportionality and pigmentation. And so it's a very, very diverse group of fishes that have resulted from this adaptive radiation. And this radiation occurred in Lake Baikal, which is this really interesting environment in Russia. It's the world's oldest, deepest, and largest by volume freshwater lake. I think it holds about 20% of global fresh water, which is a very important natural resource. What's unique about Lake Baikal is it's oxygenated down to depths of about 1,600 meters, which is very unusual for a lake environment. Typically, lakes become anoxic beyond about 100 to 200 meters. But in this case, through a combination of Ekman spirals generating downwelling currents, as well as seasonal turnover from heavy freezes, results in the carry of oxygen all the way down to the very depths of the ocean, lake. And the consequence of this is it's created a highly endemic and marine-like fauna that has supported a number of independent adaptive radiations, particularly in invertebrates. And this includes traits like giganticism in invertebrates that you might usually see in deep seas. There's a freshwater seal population in Lake Baikal, as well as hydrothermal vents and vent-adapted communities. So it's sort of this interesting freshwater replicate for looking at the types of changes that we usually see in marine environments. We've collected samples for the study in collaboration with Mike Sandel and Andy Aguilar, as well as with Sergei Krylchik at the Russian Academy of Sciences. These fish can be caught through trawling, trapping, and seine netting. We're also working with Feder Skill, also at the Russian Academy of Science, to get a high molecular weight DNA sample of this one fish in particular, which is the Baikal oilfish, which we are hoping to make a chromosome level genome assembly from. We sampled about 21 out of 33 total Baikal sculpin species, as well as 3 outgroup sculpins and 4 non-perciform outgroup, non-sculpin perciform outgroups. And the types of sequencing we've done so far is largely targeted sequencing, although again, we are moving in a whole genome direction. But we like targeted sequencing for a couple of reasons. One is that in comparisons over large evolutionary distances of tens of millions of years, much of the genome is very difficult to meaningfully compare between species because of the lack of sequence homology. And so rather than sequencing whole genomes, we've been doing targeted sequencing approaches where we look at protein coding exons, as well as conserved non-coding elements that do not encode for proteins, but are nonetheless highly conserved between species. And these often function as putative gene regulatory regions and enhancer elements regulating protein expression. And by not sequencing the whole genome, we were able to sequence a large number of species, including the 28 species in this particular data set. And we have fairly high coverage across the data set of about 95 to almost 98%, 99% of ultra-conservative elements, and at very high depth. And this allows us to track patterns of protein coding evolution as well as putative gene regulatory evolution across the history of the phylogeny. So if we make a time-calibrated phylogeny, for example, one thing that we can see is that the radiation of Baikal sculpins, which starts here, corresponds very closely with the deepening of the lake. So Lake Baikal was relatively shallow for, again, I said it's old. Most lakes aren't nearly this old, for 10 million years to about 3.5 million years ago. And then at about 3.5 million years, it reaches depths of about 600 to 1,000 meters, and then progressively deepened over time. And what we see is that it's around the time of this deepening that we start seeing early diversification of the Baikal sculpins, which then accelerates a bit as fish are migrating into deeper and deeper waters. And one thing we were curious about is in our analysis of Antarctic notothenioid fishes, we had seen this transient elevation and substitution rate genome-wide that actually returned back down to baseline during the adaptive radiation itself, and may have sort of presented an increase in genetic diversity that we thought might have facilitated parts of the adaptive radiation. We don't see any evidence for that in Baikal sculpins. So here we're looking at DS, which is the synonymous substitution rate, and it's a general proxy for substitution rate at large. And what we see is that if anything, there's a decrease compared to freshwater sculpins, although it's a highly variable thing. And if you look at the y-axis, though, there's basically no difference between any of these. We do, however, see differences in the non-synonymous to synonymous substitution rate, which is indicative of protein coding adaptive evolution. And we see that Baikal sculpins do have an increased rate of non-synonymous mutation, which probably reflects their adaptation into these deeper environments where it's cold, there's high hydrostatic pressure and low light and other physiological challenges. As I alluded to in the introduction, a hallmark of both this radiation and the notothenioid radiation is evolution from a benthic fish that spends a lot of time resting on the substrate into descendant lineages that can forage in the water column. And both common ancestors, the sculpin and the notothenioid, lacked a swim bladder, which is the air-filled sac that fish can use to hover in the water column without actively expending energy swimming. And so they became more buoyant through a reduction in skeletal density, which lightened the load, as well as through an increase in lipid content, which provides a static uplift force. And this has happened again repeatedly, both in Antarctic notothenioids as well as in the Baikal sculpins. Just to put some numbers to that, if we look at lipid as a percentage of body weight, one of the benthic sister groups to one of the pelagic sculpins is about 8.5% lipid by body weight, whereas the Baikal oilfish, true to its name, is almost 40% lipid by weight, which is frankly ridiculous. You can also see their skeleton is decreased from a baseline of around 4% of body weight to less than 2%, resulting in a massive decrease in overall body density and increase in buoyancy. These benthic to pelagic transitions evolved twice in Baikal sculpins, both in the genus Comophorus as well as in the genus Catacomophorus. And this independent evolution allows the potential to look for patterns of convergent molecular changes that might be associated with these sort of independent evolutionary events. And so we've been looking for patterns of molecular convergence, both at the site level, which would be something like a convergent amino acid, as well as in a more regional approach based on looking at whole genes or sliding windows across a chromosome, where we can look at convergent acceleration or constrained evolution that may or may not involve the exact same mutations, but could be nonetheless occurring independently in convergently evolving clades. And we're also looking at a pathway level to say, maybe if you take a gene regulatory network, maybe it's different nodes in each independent occurrence, but it all potentially clusters in the same sort of overall biological process. What we've seen so far is that there is evidence for convergent amino acids in the convergently evolving pelagic clades. In fact, there's way more of them compared to topologically similar control comparisons. suggesting that this may be associated with their convergent evolution beyond some sort of baseline level of convergent amino acids. Now, one thing we were curious about is whether or not these are, convergent substitutions, or whether or not they represent some ancestral haplotype that is then incomplete lineage sorted or shared by gene flow. So what we can do is we can look at a convergent amino acid substitution and then do gene tree windows, sort of about 5 kb on each side of that SNP, and ask if we see more than one origin of secondary pelagicism in that locus, or whether or not the pelagics cluster into one monophyletic clade. And about 80% of the time, we see that even though when there's a convergent SNP, that these fish, nonetheless, the local topology supports independent evolution of pelagicism. And so we think that this is not some broader ancestral haplotype, but is rather independently evolving in each clade, although that's something we're looking into a bit more closely. These convergent amino acid substitutions are actually enriched for things that make a lot of sense in terms of understanding benthic to pelagic transitions. In fact, these are all of the significant terms that we pulled out, and they are associated with muscle biology, which again, you're going from a benthic fish into fish that are actively swimming, a lot of collagen-related terms, as well as processes involved in lipid transport and lipid biology, which again are something that we see associated with these benthic to pelagic transitions. Here's just an example of a couple of genes. One of these, SREBF1, is a master regulator of lipid homeostasis, and overexpression leads to accumulation of triglycerides and other fatty acids in the liver. And what we can see is that they've shared this asparagine residue. And as you can tell from the broader context, they're not necessarily some shared ancestral haplotype where you have a methionine here defining this group, not seen in Catacomaphorus, for example. Other genes like LPGAT1 is associated with obesity in humans. You can see a methionine at this particular position in a region of protein that's very highly conserved. And so, it's a potentially interesting gene to look at a little bit more closely. We've also looked in a broader context on a gene basis, looking for convergent acceleration or constrained evolution. And we once again see enrichment for things associated with muscle and lipid biology. And the very top hit that we're interested in is this fatty acid elongase elavo3. And these red lines here indicate non-synonymous substitutions. And you can see that throughout the whole phylogeny, there were none in sculpins, and that these have accumulated quite a number of them. And even though some of them are at the same position, they're actually divergent substitutions, not conversion. So we think this gene might be in drift independently within both clades. What's particularly compelling about this gene is it's associated with non-alcoholic fatty liver disease in humans. And these fish in particular have extremely fatty livers of about 23% lipid by weight. And so this may be a potential genotype to phenotype connection that we're hoping to generate zebrafish models of to look at a little bit more closely. So I just want to touch briefly upon the skeleton because there's not a lot of data on it. But what we and others have shown that early in the Notothynioid, then we're back to Antarctic fishes evolution, there was a substantial reduction in skeletal density shared both with the Antarctic radiation as well as with the non-Antarctic sister group Alginops. And then this reaches extremes in some of the pelagic lineages like Disostachus, where there's even less skeletal density, facilitating their pelagic lifestyle. And so the idea was that these fish may have been primed to diversify into the water column by already having a pretty significantly reduced skeleton. Some of our collaborators, and we're not involved in this, but some of our collaborators have shown that there is in fact, a substantial reduction in the skeleton in benthic to pelagic transitions, but they have not yet looked at whether we see this early reduction in skeletal density, although based on the skeleton of the benthic sister group, we suspect that it is not in fact some early reduction in skeletal density that is then reaching extremes in the benthic to pelagic transitions. So that we've had just to kind of summarize some of the insights we've had so far, we're seeing some very distinct trends from what's happening in Antarctic Notothynioids, and that we don't see any changes in substitution rate really before, during, or after the radiation. And it's unclear if there's shift in skeletal density, for example, is occurring prior to the radiation, but it is unlikely at this point. We are seeing evidence for molecular convergence or potentially shared ancestry and lipid metabolism in the evolution of benthic to pelagic transitions, including a gene of particular interest, evolvo3, that is convergently accelerated within the secondary pelagics. And so the next steps is we're generating zebrafish models of some of these genes and mutations, and are trying to make direct comparisons between the bicoll sculpins to notothynioids as well as to deep sea marine lineages. Whereas right now we're kind of focusing within each group. The plan is to then expand and focus across to look for patterns of convergent evolution and other interesting genetic trends. And so I just want to acknowledge again, Andy, Mike, Fedor, and Sergey, who have been very instrumental in getting us some samples and have been working with us in the sort of branching out from Antarctic work into some in Lake Baikal. So with that, I'll take any questions, and thank you for your time.
Thank you very much, Jake. We have a few time for question, but I don't know if there is some question. We could take one at least. If not, I will have a question about the reduction of the density inborn in notothynioid fish. What would be the hypothesis to the initiation of this process? If not, generally we consider that because the disappearance, extinction of many fish in Antarctic, we had a lot of free niche happen and promote the recolonization of the pelagic realm. So what would be in out of Antarctica as explanation for this tendency to reduce the bone density?
So it's I don't know why, for example, Allogynops or other sub-Antarctic notothynioids would have a reduction in skeletal density, or why that would happen. That's a great question. There's a lot of interesting hypotheses for the sort of cause of this on a molecular level. One is paedomorphy. A lot of notothynioids have retention of notochords and other paedomorphic traits that might be associated with sort of premature truncation of skeletal maturation. There's also some hypotheses associated with the correlated evolution of reduced skeletal density and increase in lipids, in that mesenchymal stem cells differentiate both into adipocytes as well as into osteoblasts. And so regulating the differential fate of those mesenchymal stem cells might be important in sort of regulating that switch. But from a ecological context, yeah, I don't have a good idea as to why you might see early reduction in skeletal density within notothynioids.
Okay, Jake, is an open question, so.
That's right, that's right.
Thank you. We will move to our last talk. It's a pre-recorded, even if Abigail was here, but seems not. I will ask to Scar if they can move to this pre-recorded talk of Abigail Jackson.
Hi, my name is Abigail Jackson, and I'm a master's student with Dr. Byron Adams at Brigham Young University in Provo, Utah. Our lab studies the soil ecosystems of the McMurdo Dry Valleys in Victoria Land, Antarctica. And the title of my presentation today is Tracing Patterns of Climate with Continental Lindsay nematode. So what I mean by climate is both the historical and present day changes in climate that the McMurdo Dry Valleys has experienced. This ecosystem has remained largely unchanged as a cold desert climate since its formation sometime in the mid-Miocene, more than 5 million years ago, and it encompasses majority of the ice-free environment on the continent. This means that it has the largest terrestrial ecosystem for Antarctica's Metazoan. Even so, biodiversity is extremely low. There's only 5 nematode species, 2 tardigrade species, Rotifera, and then other protozoa. Only 65% of the soils are even habitable because of the extremely harsh conditions found here. So we have extremely low soil moisture, ranging from 2 to 5%, high salinity that can be more than 2,000 microsiemens per square centimeter. We have limited nutrients of carbon, nitrogen, and phosphorus, and then extremely cold soils ranging from -40 to 20 C, and then multiple freeze-thaw cycles throughout the season. The last glacial maximum around 7,000 to 20,000 years ago brought the largest ecosystem disturbance to this area as paleo lakes formed from the Ross ice sheet incursion that inundated large tracts of the major valley systems. So in this map here, those red zones are areas where there was inundation by paleo lakes or by the Ross ice shelf. Today, the current climate trends are warming, which is increasing the soil moisture and altering the species distributions of nematodes in the McMurdo dry valleys. The object of my study was to utilize population genetics of Scotnemellensiae to assess the historical climate change impacts on endemic species distribution, recovery, and phylogeography. So the questions I was hoping to answer with this study are: one, where is the highest genetic diversity? Are these potential origin populations? Is it in refugio sites or in recently glaciated sites or recently inundated sites? And how are these populations structured? Is it by site, by valley, proximity to the coast, maybe by assumed age? And then 3, is there gene flow among populations? Are there migration events happening? Where is gene flow the greatest? Are there source and sink populations? And then is there isolation by distance? So to do this, we sampled 27 sites that represented various geological histories from all extents of the dry valleys, from the northern, southern, western extents. We extracted nematodes, and specifically with Scotnemellensiae, we did DNA Sanger sequencing of the mitochondrial CoA1 gene, aligning 284 base pairs for phylogeographic analysis. So my first question was, where is the highest genetic diversity? To test this, I use Nye's unbiased genetic diversity index that compares polymorphisms within a population, and this is corrected for sample size. So as at the population level, we have the highest genetic diversity at these refugio lake sites mostly, and then also these 3 high elevation potential refugio sites in the squares. The difference between the genetic diversity of refugio type sites and sites that were more recently inundated during the last glacial maximum are actually fairly similar though. Next, to look at how populations are structured, either by site, valley, assumed age, I use an analysis of molecular variance to determine how populations are structured. This test uses an FST, or genetic differentiation calculation, that evaluates the proportion of genetic diversity among a population versus the total population. So what we see here on this first graph is that within population, there is significant genetic distance. So each population is genetically distinct from the total sampling sites. The second graph shows that differentiation of populations in the same valley system are actually not that significant. So there must be some gene flow occurring within valley systems. And then lastly, there was significant differentiation between valley systems. So there is limited gene flow across the mountain and glacial ranges that separate valleys themselves. We can visualize this pattern in a haplotype network. So here we have it colored by populations. There is a large genetic distance or number of mutation events that's separating these northern and western valleys. So Alatna in red, Wright Valley in blue, McClivey Glacier over here in green, and then Victoria Valley in this gold, and Wright Valley in blue, all indicate a pattern of long-term isolation. Meanwhile, these more coastal southern valley sites, so Taylor in pink and purple and blue, and then Mears in this yellow and Garwood in this turquoise color, they appear to have a lot more gene flow and recent expansion event given the presence of 2 main haplotypes with lots of satellite haplotypes only a few mutations apart. This can also be represented here in a dendrogram. Now we're looking at just the valley systems here. So we notice a well supported by 1,000 bootstrap monophyletic clade in Taylor, Mears, and Garwood at the bottom of this dendrogram. They all have short genetic distances between these valleys, whereas on the top of the tree, the long-term isolated northern and western valleys, they represent their own monophyletic clade with more genetic distances between each valley. So my last question was if there is gene flow among populations. So testing this required running an isolation by distance model that compared the geographic coordinates and genetic distances of Nye's Euclidean distance against the null hypothesis of genetic distances and geographic points that did not have any isolation. So the full data set shows a strong significance of isolation by distance. And the next step I will take is to test isolation by distance within valley systems, maybe to evaluate gene flow at population levels here, and also gene flow between those Mears, Taylor, and Garwood sites that showed less genetic diversity or less genetic distance between them. So to wrap up, I hope I answered these questions of where is the highest genetic diversity? Genetic diversity was equal for potential refugia populations and more recently inundated sites in terms of this ecosystem. So high genetic diversity at the low elevation, recently inundated sites can maybe be explained by increased connectivity from either cryoturbation as soils flow down from the surrounding peaks, even down valley katabatic winds, ephemeral streams, and lakes. Those would help facilitate migration. The higher elevation sites, they have a little bit less opportunity for migration. There's no water or down valley winds. So migration on a large scale is a little bit more limited. That makes their genetic diversity a little bit more interesting, as it might be an artifact of more long-term isolation and genetic drift. Number two, our population structure. Yes, the AMOVA test clearly found significant structure on a valley basis. And then three is their gene flow over the entire population. The isolation by distance model can be interpreted to mean there is limited gene flow, and then it'll be interesting to test populations within a valley and then valleys that were forming monophyletic clades. So in conclusion, this project demonstrates phylogeographic patterns left from different geological and climate histories. As populations are highly structured and gene flow is limited for Scatinema lindsay, it's showing that this nematode responds really slowly to disturbances, which under future climate change may have disproportionate impacts on carbon cycling, given this nematode's dominant role in this low diversity ecosystem. So with that, I thank you for listening for this last presentation, and I'll take any questions. Thanks.
Okay. Okay, we'll thank Abigail. We can put off the video. She's not here to answer a question. She was here at the beginning of the session, but we will put yes, we have the email of Abigail in the chat for people. I think it's really important where we have seen here in this two-session some advance in terrestrial genetic diversity. We have plenty of examples studying for marine species, but very few for terrestrial. It is now increasing, and we saw also see some phylogeographic study for bacteria. So it's also great news for this discipline. So we would like to thank all the speakers and all the participants of this session. I will just say to you that we have around 20 posters for this session. You can have a look to this poster at any time during the Congress. So thank you very much for all people that assist to our long session. Thank you very much.
Thank you.
Bye.
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 Thank you.